start, Musa? Hello, dear students. Today we have to discuss two very important chapters for NEET, that is plant growth and development and cell cycle and cell division. Okay, so if you will look at the previous year questions of these two chapters, you will be able to see that they will be asking so many questions from plant growth and development as well as cell cycle and cell division, in which cell cycle is the most important chapter, right? But what we'll do today, we'll complete plant growth and development and then we'll go to cell cycle and cell division chapter. Okay, because it is actually very easy chapter, it will take less time, right? So let's begin plant growth and development. So on the slide, you can see that we have given the most important points from the chapter, like which are there in NCRT, right? So we'll revise all those points together and we'll be solving some questions based on these points only, right? So you can see that when we start this chapter, they have mentioned that all the living organisms, whether it's a plant or an animal, we all have started our life as a single cell, that is called zygote. So we can say that all the cells are descendant of a single cell, that is zygote. Can you tell me what is the ploidy of zygote, right? Now, development is the sum total of two processes, right? Up, let's suppose a plant is growing, right? So there are so many stages which a plant has to complete, then only it can develop into a plant, right? So that development process includes what all things? Development includes growth and differentiation, right? Growth has two meanings. You have learned this from class 11 living word chapter itself, that growth can be considered as increase in the number of cell, that is called cell division, right? Because of cell division, you can see number of cells will increase into any kind of organism. And cell elongation, after division, every cell must elongate, right? So that it can acquire its particular size. And after completing cell division and elongation, or you can say growth of a cell, for a cell has to differentiate. Differentiate means the cell has to mature. A cell has to you can say become specific for performing a specific function. This is called differentiation, right? Or cell maturation. Only because of these two processes we can see a plant or an animal or any kind of organism will develop. But because we are reading about plant, we have to consider plant here, right? So plant show development because of growth and differentiation, which occurs at cell level, and as an entire organism you will see that plant will be developing altogether, right? Now here in NCRT they have given certain steps which you have to remember. They have given that whenever a zygote converts into a mature plant, there are some ordered strict series of events which are going to occur, right? For example, the very first thing you have to remember, all these you can say events, or you can say all these phases, they are very much precise, they are highly ordered, we cannot change them, right? And they will occur one by another, means next to next, right? For example, whenever you will see a plant growing or developing, you will see, you will germinate a seed. Seed germination will take place. A small plant will come out, that is called seedling. That plant will enter into its young phase, that is called juvenile phase or vegetative phase. Then the plant will undergo maturation. During maturation you will see flowers will appear, right? Flowers are the sites where sexual reproduction is going to take place, and as a result there will be formation of seed, right? And then what will happen, as soon as a plant will bear seeds, it means that the plant is entering into another stage, that is called senescence. Senescence is what? It is a stage of a plant which leads to death of a plant, right? So it all started from seed germination, this is the first step. Then you can see it is ending at senescence, that is the last stage of the development, right? So these are some of the stages of development which you have to remember. They are very precise, they are ordered, and they will occur sequentially, step by step. Now here you can see this development process, it has multiple stages I have mentioned. And when you see complex body organization, like in the plant, when you will see there will be formation of certain vegetative structures like root, leaf, stem, even the reproductive structures like flowers, birds, fruit and seed, right? All of these things are formed because of these precise ordered sequences, right? And it is affected by multiple factors, right? So plant is developing, plant is producing so many different structures, plant is growing. First it is entering in juvenile phase, it is making leaf, right? It is elongating the stem, it is developing the root system. After that it will be forming the birds, flowers and all, right? So all this happens because of many factors, right? That can be plant's own factor, right? And it can be affected by certain environmental factors as well, right? So you have studied about plant own factors mostly in plant in photosynthesis chapter, right? There are so many factors which affect the growth of plant, right? Like what, how many leaves are there, what's the color of leaf, what is the size of leaf, how much chlorophyll is there inside that leaf, right? What is the oxygen amount which is present in the plant body, what is CO2 concentration which is there inside the plant body. So many things are there which all together are related with the plant itself. What are the hormonal secretions which are there, right? Or hormonal synthesis, right? This all affects development of plant. On the other hand, there are certain environmental factors, environmental conditions which also affect development of plant. For example, if the conditions are favorable for seed germination, then the seed will definitely germinate, right? The first process of development will be checked. It will germinate if conditions are favorable, right? But if the conditions are unfavorable, then the plant will not develop on at that time, right? It will enter into a state of suspended growth, that is called dormancy. During this period the plant will not grow at all. The plant will wait that when the conditions will become favorable, at that time only the plant is going to germinate, right? So what is this germination, how it is affected by different environmental factors? Can you tell me? In class 11th you have studied for this seed germination, right? You require certain specific things. uh, what are those things? Remember, you have studied that a seed requires a suitable temperature, right? If this suitable temperature is not there, then the seed is not going to germinate, right? And then you have studied that it requires oxygen, right? If oxygen is not supplied, then also seed will not germinate. What is the use of oxygen for germination of seed? Can you tell me? You remember, you have studied that within the seed embryo is present, that embryo must be growing into a plant, right? Or embryo has to change into seedling stage. For that the embryo has two apical meristems. What are the names? Shoot apical meristem and root apical meristem. And these meristematic cells need to divide, then only you will see growth occurring, right? And for division, for cell division, what is needed? Energy, right? From where energy comes? From aerobic respiration. For aerobic respiration, what is required? Oxygen, right? So this you have to remember from other chapters as well. And along with suitable temperature and oxygen, what else is required? You remember, we require adequate amount of moisture. Where you have studied this? You have studied this in class 12th, right? When seed is there in the topic, in the chapter sexual reproduction flowering plant, there you have studied these 3 points. Okay, so if you have these conditions which are favoring for seed germination, then seed will germinate. If moisture is unavailable, oxygen is not enough, or temperature is not correct, not suitable for seed germination, seed will enter in a state of suspended growth, that is called dormancy, right? It will wait that when the conditions are reversed back to normal, then only it is going to germinate. Now just tell me, what is that particular cell with which we all started our life? Or you can say all cells of plant cells are descendant of, right? So you know the answer very well. The answer here is zygote, right? This kind of question they will ask for 4 marks. Development consists of what? Development consists of growth as well as differentiation. So both A and B will be the answer. Just now we have studied this, right? Then which of the following is correct about the development of a mature plant from a zygote, right? If you want to develop a mature plant from a zygote, then which is correct? It will follow a precise and ordered succession of events. Only differentiation occurs due to environment effect only, right? So see, more or less whatever they have written is correct here, but the way they have expressed the expression, like statements are not correct, right? Just we can say that yes, if we are talking about the development of a mature plant from zygote, definitely environmental factors will affect it, but only environmental factors will affect? No, that is wrong, right? Only differentiation is going to affect? No, differentiation definitely will affect development, because growth plus differentiation means development, right? But only and only differentiation? No, right? So what we should mark? We should mark this one, that it should follow precise ordered succession of events, right? First seed should germinate, seedling should be formed, right? Juvenile stage should appear, then reproductive stage should appear, then flowering should take place, right? Then seed formation, and after that senescence. So it's a ordered sequence which you have to remember. What is the first step in the process of plant growth, right? So when a plant is growing, right, what is the first step? It is that the seed should germinate, the embryo which is present within the seed should germinate, right? So you can see from a very small topic itself we can have a lot of questions, right? Now here you can see one important diagram which is given in NCERT. Here they have mentioned germination and seedling development in bean, right? Bean is dicot or monocot? Bean is a dicot plant, right? It belongs to which family? It belongs to Fabaceae, right? Now they can ask a question here. They can give this diagram to you. You have to remember the labeling because nothing else is mentioned for this diagram. So if they want to ask any question, they can ask you where is hypocotyle, where is cotyledon, right? So you have to remember this. You know the meaning of hypocotyle and cotyledon, right? There is an embryonal axis, right, which you have studied in morphology also, and in sexual reproduction in flowering plants also. So the body of embryo is divided, or you can say the body of embryo is planned on an imaginary axis. That imaginary axis is called embryonal axis, right? The part of the axis which is situated above the attachment of cotyledon, it is called epicotyle. The one which is situated below the level of cotyledon, it is called hypocotyle, right? So there are different type of germination where epicotyle will come out first or hypocotyle will come out first, right? But that is not given in NCERT here in this chapter, right? So you just have to remember this portion is epicotyle, this is hypocotyle. In each and every diagram you can see they have mentioned. Here they have shown this is as hypocotyle, right? This as hypocotyle. Here hypocotyle is uh, not coming out first in germination, epicotyle is coming out first, right? And here what is happening? Here hypocotyle is coming out first in germination. So this diagram labeling only you have to remember. You don't have to remember what kind of germination it is, epigel or hypogel. That will not be asked here, right? So you have to remember the labeling. Now the next part of chapters talks extensively about growth, right? What is the meaning of growth as per this chapter? So you know very well that growth is an irreversible permanent change in the size of an organ or its part, right? Or individual cell or the entire plant body, right? We are reading about growth and development in plant, right? So you will have to remember that if a plant is growing and the change which is occurring is irreversible, right? The size of an organ is increasing, the part of a plant is increasing, or individual cell of a plant is increasing in size, or entire plant body is increasing. This all can be considered as growth. Okay, just say for example, can I write that expansion of leaf is considered as growth? Yes, because what is the meaning here? Size of an organ is increasing, right? Can I write here that stem elongation is growth? Yes, because the length of the stem is increasing, right? It's an organ of angiosperm. So if an organ of plant is increasing, or or entire plant is increasing, or individual cell of a plant is increasing in size, that all can be considered growth. Can you reverse it? No, it is irreversible, it is permanent change, right? Now whenever growth is occurring, it is accompanied by two processes, right? What are those two processes? Those two processes are both anabolic process and catabolic process. So altogether we can say that growth is accompanied by metabolic process, because metabolic process includes anabolism and catabolism. What's the meaning of anabolism? Synthesis of something. What's the meaning of catabolism? Breakdown of something, right? So just imagine that if a plant cell is growing, right, it has to synthesize so many things. At the same time, it requires energy also. So it has to do that catabolic pathway of respiration as well, aerobic cellular respiration, right? So you have to remember, because we are talking about growth, we have to make something. So here anabolic processes will always be more in number, right? We will have catabolic processes also, but as compared to anabolic processes, they will be limited only, right? Because this will only give us energy. This will be responsible for synthesizing everything. Let's suppose a plant cell is dividing and making a new plant cell. Then what all things are required? Everything is required, right? You have to copy everything. Every cell organelle must be copied. Nucleus must divide. DNA must divide within that nucleus. All the endoplasmic reticulum, Golgi complex, they must be duplicated. So you have to synthesize this, duplicate this. For that you require energy, right? But you have to duplicate more, much more things as you require energy, right? You need more plasma membrane, you need cytoplasm amount to increase. Lot of changes are needed. Cell wall synthesis is required, right? So for synthesizing things, always remember, growth may anabolism activity will be higher always as compared to catabolism, right? Then in plants specifically, the growth is always localized. What is the meaning of this? Means that in plants, growth will occur only at specific regions. Plant will not show growth from each part and every part, right? It's only having some specific locations where you will see growth is occurring. And you know very well what's the reason behind this. Reason is that plant body is divided into different type of tissues. For example, permanent tissue and meristematic tissue. So permanent tissue ke place se it is not dividing normally, right? Plant is showing us division only from the growing parts, that is their meristematic parts, that is most of the times situated at the tips, right? But you have other types of meristems also which you have to remember. I'll show you one figure for that. Now it is unlimited, growth is unlimited, right? Plant will keep on growing, it will continue to grow, it will not be like us, right? We have a limited type of growth, plants are growing unlimited. Why? Because they have meristematic cells in their body. So here the growth is going to occur throughout the life, right? And you can also say that this growth is indefinite type, right? Indeterminate type, right? But for individual organ of a plant, just like you can take an example of a leaf, right? The leaf or organ, right? Maybe any kind of organ you can take. For simplification, I'm taking leaf only. If you consider at organ level which type of growth it is, so it must be definite growth, right? You will never find a plant which is having one leaf of this size, another leaf of this much size. No, right? There must be some, you can say, fixed type of size. In that range only the leaf or maybe the stem it will grow. But can a plant grow throughout its life? Yes, because it is having meristematic region, so its growth is unlimited. This kind of growth where you can see that the growth is continuous throughout the life, it is called open type of growth, right? New cells are always added up to the body, right, which always become differentiated, right? So this kind of growth only exists in plants, not in animals. So growth is localized, growth is unlimited, it is of open type. And can you measure this growth? Definitely you can measure this growth. Growth is measurable, right? Now I'll show you the image which is given in NCERT, the diagrams which you have to consider, and you have to remember the labeling, the heading, everything, right? Under heading, everything you have to remember here. You can see that here they have given us a plant and they have represented certain meristems which you have very nicely studied in anatomy of flowering plants, right? And in the next session we have to revise that chapter. Now see, here at the tip you can see they have mentioned these cells are called shoot apical meristem, right? Here these cells, they are going to, this arrows are denoting that the daughter cells which are formed, they will be added towards the downside, right? So here when new cells are added here, the permanent cells, definitely you will see this structure will elongate, right? It will grow like this in the ascending direction, right? So the arrow is showing you what is the position of daughter cells, where the daughter cells are going, right? And this is showing you the dividing cells, right? Then here also you can see this is the root apical meristem at the base of this diagram, and you can see the daughter cells which are added are adding up to the upper side. So here the growth will occur in this direction, that is called descending direction, right? It will grow like this will go up like this because of that daughter cells where they are moving, right? And then you can see they have tried to show us the circular type of meristem, that is vascular cambium here, right? They have shown vascular cambium only, but you have to remember, along with vascular cambium we also have one more cambium here, right? What is the name of that cambium? It is called cork cambium, right? Vascular cambium is also there in circular fashion. It is there, it is present like this here, sorry, here, and the outermost you can consider it is cork cambium, right? Together both of these meristems, what they make, they will make secondary meristem, right? Origin wise they are secondary meristem, function wise also they are secondary meristem. In the next class I will explain you. And this shoot apical meristem, it is primary meristem. Root apical meristem, it is primary meristem. It is present in embryonic stage of plant as well, right? So you have to remember these labelings of the diagram and what they want to show here. This diagram represents the location of root apical meristem, shoot apical meristem, and vascular cambium only, right? They have not shown here cork cambium, but you should know that it is present there. But if asked in question, you have to remember only three names, right? Shoot apical meristem, root apical meristem, and vascular cambium, right? And the arrows, they have mentioned very clearly, arrows are exhibiting the direction of growth of cells and organs. So the growth will occur in this direction here, and the growth will occur like this, right? It will increase the width. Remember, if you have a vascular cambium like this, new cells will be added in this direction also, this direction also, right? As a result, the girth of this particular plant will increase. So they want to show these arrows to make sure that the girth is increasing, right? So this diagram you have to be very careful. Now this diagram is also given, about which they have not explained anything. So just you have to remember that this is the seedling which germinated and elongated further. So this is showing you zones of elongation, right, by parallel line technique. If they ask this question, only one question they can ask, this is you can say find the elongation by which method. So you have to remember, by parallel line technique, this method has been used to understand the growth of, you can see now they have drawn parallel lines. So up to here it's normal, and then you can see it is increasing in length, right? So by making parallel lines we can understand how much elongation has happened in a germinated seedling, right? Now I told you that growth is measurable, right? But actually, what is real growth in plant? Real growth is increase in the protoplasm, right? If I'm able to say that yes, an entire plant is growing from this much size, the plant has become of this size, right? So actually, what is happening and how it is occurring, how it is happening? So it is happening because of individual cell ka protoplasm is increasing in size, right? So if you want to measure actual growth, what you have to measure, you have to measure actual protoplasm increase, right? Which is not possible, which is very difficult job, which is very tedious job, right? We cannot measure the increase of protoplasm in individual cells of the plant body, all the cells of that plant body, right? It's not possible. So what we have to do, we have to find some alternate ways by which we can measure growth, right? And those ways I'll tell you. Before that, there's one question which you must answer. Tell me what is the answer of this question. It's the most easy question which you can do. In the beginning I have taken all the questions which you can do very easily. So you know very well, irreversible permanent increase in size, right, of an organ or its part or even individual cell, it is called growth, right? This is the most easy question. If there is expansion of leaf, right, then what you'll call it? You'll call it as swelling, or you'll call it as imbibition, or differentiation, or growth. So you will be calling it as growth because it is increase in size of a plant organ, right? Now, assertion reason question. Growth in plant is open, right? It is open. What is the reason? New cells are always being added to the plant body by the activity of meristem, and the cells or tissues arising from a given meristem has similar structure and function at maturity, right? So what is the reason they quoted here? The reason they have quoted is that new cells are always being added to the plant body by activity of meristem. This is so correct. But the cells or tissue arising from a given meristem has similar structure and function at maturity. If you read this statement, do you think this statement is correct? No, right? They have given that the cells and tissues which are made by this meristem at maturity, they all will be similar structured and they all will perform similar function. That is wrong, right? You have to remember one thing, right? See, if we have a meristematic cell, right, and this meristematic cell, okay, in plant body, whatever permanent cells which we are able to see, they all are derived from meristematic cell only that dividing cell will divide and give rise to daughter cells which will become permanent, right? So meristematic cell will differentiate, right? Here what will happen? It will divide, followed by its elongation, right? Then it will differentiate. This is what we have studied in development, growth and differentiation. What is the meaning of differentiation? Differentiation means it will become specified to perform a specific function. Differentiation can be of any type, right? For example, if I am saying that I want to make different type of cells from meristematic cell, right? Let's suppose meristematic cell divided, and here I want to make a cell which is mesophyll cell. Here I want to make a cell which is epidermis cell. Here I want to make a cell, let's suppose, which is, let's suppose, any cell of xylem, maybe trachea. So you tell me that these all are different type of cells, right? All these different type of cells have different size, right? Different functions they are able to perform, right? But they are formed from same meristematic cell, so they should undergo different type of differentiation and different type of elongation or not. Understood what I'm trying to say? So mesophyll cell, if I want to make from from a daughter cell of meristematic cell, then I should have lot of chloroplast in that cell. I should have high pigment concentration in those chloroplast, right? I should have all the enzymes necessary for photosynthesis, then only it can do photosynthesis. If I want to make epidermal cell, then what I should have, these cells should be devoid of any kind of chlorophyll, right? They should not have any chloroplast. All the epidermis cells are non-green cells, right? They should be located on the outermost part of the plant body, right? There should not be any intercellular spaces between them, right? But mesophyll cell can have, or maybe it is missing, right? Trachea should be elongated, it should have sorry, it should have lignin containing cell wall, right? Understood. And it should be losing all the protoplasm and it should become dead at maturity. So what is the meaning here? Different kind of changes will occur here, right? In this differentiation, different changes will occur, and you are going to get different type of cells from a same meristematic cell, which are going to perform different functions. Function of these cells, which, or you can say the tissue which these cells will make, they all will be different. Okay, so saying that the cells or tissues arising from a meristem has similar structure and function at maturity is absolutely wrong, right? So what we have to remember? We have to remember that the assertion which they have quoted is actually correct, right? It's correct assertion, but the reason they have quoted is half correct only. Up to here if they have given, it's fine, but the other half is wrong. So what we have to choose? We have to choose that the given assertion is true, but the given reason is false. That is option C. Are you able to understand? Right. Now, if you cannot measure growth by measuring the amount of protoplasm which is increased, then you can measure it by some alternative method. What is that alternative method? There are different type of parameters which you can use. Just say for example, you can use increase in fresh weight, right? What is the meaning of increase in fresh weight? You all have studied the chapter where they are talking about the biomass production, right? So you have studied this in previous junior classes also, and you have studied this in ecosystem chapter as well, right? So fresh weight and dry weight are the two things which denote what is the biomass of the plant, right? Biomass of a plant or total weight of a plant can be calculated along with the water content of the plant, that is fresh weight, and without the water, which is only the carbon content of the plant, that is dry weight, right? So for fresh weight, you have to take the weight of entire you have to weigh the entire plant along with the water, right? As it is, you can weigh it. And if it is dry weight, then you have to take the plant, dry it up in oven, and then only the carbon part ka weigh you have to calculate, right? That will give you increase in dry weight and increase in fresh weight. अब increase के लिए क्या करना है? You have to take final weight and you have to take initial weight, right? And final में से you have to minus that initial weight and you can get how much increase has occurred, right? So increase in the fresh weight minus sorry the fresh weight, okay, initial final fresh weight minus initial fresh weight, you will get increase in fresh weight, okay. Final dry weight minus initial dry weight, you will get increase in the dry weight, okay. And what is fresh weight, dry weight? I told you it is method of calculating biomass with water, without water, only carbon here, okay. Why we are interested in knowing only carbon ka weight? Because photosynthesis mein carbon dioxide is fixed. So how much carbon dioxide has been fixed, that can tell you what growth has taken place. Now if you want to calculate growth in terms of increase in length, you can do that. For example, certain cells are there, like pollen tube formation occurs in pollen grain, so you can calculate the elongation or increase in the length of a pollen grain by checking the length of pollen tube, right? If you want to calculate length for the organs like leaves, you can rely on surface area, right? Increase in the surface area. Then for fruits, you can use increase in volume, right? And or you can individually you can calculate what is the increase in number of cell. So these are some different parameters. If you cannot do increase in protoplasm, then you can use these parameters for calculating or measuring growth, right? Then there's one device which is called oxanometer. This oxanometer is an instrument which measures the growth of plant in terms of increase in length, right? So this also they can ask, but it's not given in NCRT, right? Oxanometer, it is used for calculating the increase in length of a plant. It's a device, it's an instrument. Now, in NCRT, they have given certain statements which you have to remember, because there are so many statement questions as we can see, right? So they can ask a question here. Just they have given one single root apical meristem of a plant. Okay, it can produce more than 1 lakh, sorry, more than 17,500 cells. So one root apical meristem is dividing, and it is capable to give rise to, okay, 17,500 cell per hour. In 1 hour or 60 minutes, it can produce, one root apical meristem can produce 17,500 cells. Then they have given that if you consider what, if you take watermelon, right? So watermelon ka individual cell can increase in size, okay, by up to 3,50,000 times, right? So these two values are given for watermelon, because watermelon is already big, right? So for that big watermelon, you have the bigger number, that is 3,50,000, right? And root apical meristem, it's a tiny meristem, meristematic cell, but in an hour, how many number of cells it can produce? It can produce 17,500 cells. They can interchange the values, and they can put a statement question to you, right? Now this kind of question directly also they can ask, that if one single maize root apical cell, right, maize is also given in CRT, maize ka root apical cell or root apical meristem is dividing, how many cells per hour can be produced? So it will be, I have mentioned here, it should be more than right? So what you will choose here? You will choose option number B. See, this is tricky question, right? They have mentioned 17,500 also, less than 17,500, right? So you have to remember more than 17,500 it can produce. Okay, now we have to study about phases of growth, right? When a cell is dividing, what are the phases in which that cell will enter to show growth, right? So this is very, very easy. You have already studied this in the chapter morphology of flowering plants. When the cells are dividing at the root apical meristem region, where the cell will enter next is region of elongation, right? Matlab, the state of cell after division will go into elongating state. It should increase in size, right? Then it will enter into region of maturation where the cell is going to differentiate. So same thing is there here as well, right? Whenever a cell is growing, right, there are three different phases of this growth. First is called meristematic phase or formative phase. In this meristematic phase or formative phase, what is the characters which a cell holds, right? So the cells which are present in this state, they are constantly dividing, one cell making two cells again dividing, right, making four cells. So they are constantly dividing. This constant division occurs because of very important properties which these cells hold. What are those important properties? The important properties which these meristematic cells, dividing cells, they hold, they carry, that is that they have a very dense protoplasm, right? They have lot of dissolved solutes, so many proteins, enzymes, everything which is needed for division is there in their cytoplasm. So the protoplasm itself, the living part of the cell, becomes dense, right? It's having a very large, conspicuous, or you can say prominently visible nucleus, because the nucleus is going to divide. I have written nuclei because I'm talking about meristematic cells, plural, right? Now, the cell must be metabolically active, right? So many metabolic reactions are occurring. Just now I told you growth is accompanied by two things. What are those two things? Can you tell me? It is anabolic reactions and catabolic reactions, right? So much is occurring inside the cell. It is a metabolically active cell. Then, cell wall should be of which condition? Cell wall should be thin because a cell has to divide. If it's having very thick, very tough, right, very strong cell wall which is not even elastic, then a cell cannot divide, right? So for division process, the cell wall should be thin. It should be primary cell wall, cellulose containing cell wall, right? And it should have lot of plasmodesmatal connections. Plasmodesmatal connections should be there with other cells so that lot of nutrition can be transported in these cells. They are not having any kind of chlorophyll, right? So they are dependent on the cytoplasm of other cells. Maybe other things are passing, enzymes are passing, glucose is passing, right? All these things. Then vacuole is absent, right? There is no vacuole here. Okay, these are some of the important points which you have to remember. Because of these characters only, the cell is in the meristematic phase. The cell is able to divide because of the characters which it carries, right? If any characters change, the cell will not be able to divide, right? Then after completing the division, the cell has to elongate in its size. It has to acquire its perfect size. So for that, what is the condition? Now proximal to this region will be there where cells will shift. All the cells after division, they will shift, right? And there you will see individual cells how they will elongate. For elongation, there will be vacuole formation. The vacuolation will increase, right? When there will be increased vacuolation, you will see visible cell enlargement. Because of this, cell will enlarge. There will be deposition of new cell wall material, right? So new cell wall material will be deposited, because imagine if a cell is increasing from this size to this size, the cell wall also will increase, right? So the cells should synthesize the cell wall material and it should be going out and deposited there, right? And there will be one enzyme which will be helping in this, that is called expansin. There will be an activity of an enzyme which is expansin. This will hydrolyze the cellulose microfibrils, right? And because of this, what will happen? The cell wall will become loosened. Imagine a condition that there is a cell like this, right? And here the cytoplasm is increasing in you can say size. Why? Because a vacuole formation is occurring here, right? So because of that vacuole formation, there will be a pressure which will be created, right? There is a vacuole formation here. So because of that, what will happen? Vacuole is now formed like this, and this is the cell wall area, right? So there will be pressure created here. Then what will happen? There is one enzyme that is called expansin enzyme. This expansin enzyme will go and it will hydrolyze the microfibrils of cellulose, right? If these microfibrils are hydrolyzed, they will become loose. If they will become loose, what will happen? There will be some space which will be created, right? And then that space will be filled by new cell wall material deposition. Means this cell will make the new cell wall material and will be deposited, and you will see the cell wall getting transformed, right? This you have to remember. Expansion, although is not given in NCRT, but actually increased vacuolation and cell enlargement with the new cell wall material deposition, these terms are there in NCRT. Then phase of maturation. Okay, after elongating, the cell will enter in the phase of maturation. This is also just near to elongation phase. Here what is going to happen? Here the cells are going to change its structure, right? Either they can change their shape further, or they can change their size. Up to here also this can take place. There will be internal constitution change, right? Internal means cytoplasm, there are so many things which can change, right? And there can be change in physiology also. So either it's a structural change or physiological change. Because of the changes in the structure, it can do particular specific function, right? After completing this maturation or differentiation, further there will be no change in the cell. The cell will become fixed, right? Cell will become fixed for serving a role, right? Further it will never change. Maximum size of the cell, maximum increase in size of the cell, uh, maximum changes in the protoplasm, that all will take place in the phase of differentiation. After that, further there is no change. Okay, this also is given in NCRT or remember. Which of the following statement is not correct? This has been asked in previous NEET, you can see. Okay, it is not correct here. Phase of cell elongation of plant cell is characterized by increased vacuolation. Just now we have seen, yes, elongation occur because of increased vacuole formation. This is true. Cells in meristematic phase of the growth exhibit abundant plasmodesmatal connection. Just now we have written, yes, meristematic phase cells cannot do photosynthesis. So if any kind of transfer of nutrients have to take place, that will be through plasmodesmata. So this is also correct. Plant growth is generally determinate. This statement, just tell me, is this true? No, right? Plant growth is indeterminate, right? It is indeterminate. But for a specific organ, to some extent we can say yes, it is determinate, but for an entire plant, no, it is not determinate, right? Plant growth is measurable. Can you measure plant growth? Yes, you can measure it. But what is the actual measurement? Increase in the amount of protoplasm. But can you measure it very easily? No. So can you rely on other parameters? Yes, right? And we have seen a list of parameters NCRT has provided us. Now, at the cellular level in plants, growth is consequence of, right? If you see at cell level, right? So growth is because of what? You can see very easily in NCRT they have mentioned increase in amount of protoplasm, right? So you have to go with word to word like NCRT. You cannot choose increase in amount of cytoplasm, right? Because cytoplasm is only that part, that semi-fluid part of the cell, right? Protoplasm includes all the living component of a cell, and if you want to grow a cell, you need to increase entire living component of cell, right? Fine. So it is increasing protoplasm. Now, you have to remember, I told you already that the maximum size, maximum cell wall changes, cell wall thickening, maximum modifications in the protoplasm will be achieved, okay, in which zone? In which zone? It will be achieved in zone of maturation. Okay. After that, if a question is asked that maximum size, wall thickening and protoplasmic modification is achieved at the end of Zone of elongation, zone of maturation, right? Zone of division. So you have to remember it is zone of maturation. It's also one of the PYQ. Now we have an idea about growth. Now we have to study growth rate. What is the meaning of growth rate? So it is the increased growth per unit time, right? It's a rate, how much fastly growth is occurring. That is called growth, right? Or how much slow growth is. That is also considered as growth. So increased growth per unit time. Okay, here they are saying that what is the increased growth per unit time, right? With a given time, how much growth is occurring. That is called growth rate. Rate of growth can be expressed mathematically, right? You actually, if you have studied this chapter, you may remember that there are two mathematical equations which are given, right? So you can express the growth rate in mathematical expressions. Now new cells can be produced in variety of ways. Definitely growth means production of new cells. After that it will increase the size of the cell, then it will allow differentiation of that cell or maturation, right? But the prime thing is that we have to increase the size, sorry, we have to increase the number of cells. So scale here, there are different ways by which we can increase the number of cells. Two important ways are given. First is arithmetic, another is here geometric, right? So here I have given a brief account of both. You can see on the board I have mentioned arithmetic growth here. This is a type of growth which occurs at a constant rate, right? There is no fluctuation in this growth. Just say for example, if you see this diagram, for many students this diagram is a big problem, right? You will not kind of understand what they want to tell here. So you have to remember they have given two cells here. One is this dark blue cell, another is like very light blue, right? So this cell, you have to remember, this is meristematic cell. Okay, this cell is meristematic in region. Okay, meristematic cell. Okay, now when this meristematic cell is dividing, it is giving rise to two daughter cell. One cell is again dark blue, another is light blue, right? So this dark blue remains meristematic, but the light blue has become permanent cell, right? Again this meristematic cell will divide, one will remain meristematic, two cells will become permanent cells. Again only one cell is dividing, one it is keeping as meristematic cell, three are becoming permanent cells, right? Here again this is dividing, this is remaining meristematic, and the other, okay, during this division this cell is added, during this division this cell is added, during this division this cell is added, during this division this cell is added. Matlab only one new cell is being added every time. Okay, the rest are coming from the previous generation. So what you have to remember here, you can see here there was one cell, then it was two cells, then three cells, then four cells, right? And then finally you can see five cells. Matlab in every step can you see one cell is adding. This is a constant growth or not, right? This is a constant type of growth where you can see growth occurs at a constant rate every you can say generation. One new cell is adding, right? This is achieved when meristematic cell divides, it will give rise to two daughter cells. One daughter cell remains meristematic and another daughter cell becomes permanent. Only then you will achieve arithmetic growth, which is of constant growth rate type, right? So same thing I have mentioned here, mitotic division is occurring, right? And only one cell is adding up in every generation. And this kind of growth, where is it is visible in the plant body, you should think that where in the plant body we can see this kind of growth. So just imagine that the tips of shoot and the tips of root, if they have meristematic cells always present there, then the daughter cell which are formed out of those two daughter cells, one should remain meristematic, another should become permanent, right? Otherwise what will happen, all the meristematic cells, if they are dividing and becoming permanent, meristematic cells will be lost or not, right? So this kind of growth, arithmetic growth, occurs at the tips, in the apical meristems, in the shoot apical meristem and root apical meristem. And for what purpose this arithmetic growth rate is used, it is there for increasing the length of the plant. For elongation, this type of growth rate is required, right? Then we have here geometric growth. Geometric growth may you can see a different kind of diagram is given, right? Here you can see here growth does not occur at constant rate like this every you can say time one cell added. No, here you can see only dark blue color cells have been drawn. It means all of these cells are meristematic in nature, right? So one cell divided and it gave rise to two cells. Both of them are meristematic. They both will divide and they will give rise to four cells. Again, these all four cells remain meristematic. They will give rise to eight cells. All the eight cells can divide and they will give rise to 16 cells, right? So in this, what you can consider, you can consider that every time the growth is occurring, it is not same, right? Here from one cell we made two cells, so here total one cell is added. But from 2 to 4 when we made, here how many cells are added? Two cells are added, right? From 4 to 8 how many? Four cells are added. If from 8 to 16 we are going, eight new cells are added. So can we say this is a regular type of growth or constant type of growth? No, right? Constant matlab fixed number of cells are added every time. Here first time it is 1, then 2, then 4, then 8, then 16, like this, right? So this is growth which does not occur at a constant rate. Actually you will see this kind of growth in cells in tissue level, right, in organ level, and in the complete plant level. But at specific regions for elongation of the plant, you have this kind of growth. At cell level, tissue level, organ level, and plant level, you have geometric type of growth, right? So you have to remember this. Now every cell here is dividing into daughter cell, and daughter cells all are dividing again. When you will plot this on a graph and you try to analyze it, you'll get this kind of graph, right? Can you see with respect to the time the height of the plant is increasing at a constant rate? So this line is denoting what? Constant rate, right? Arithmetic growth. And if you derive one equation for this, it will come as lt equals to l naught plus rt. What is the meaning of lt? Lt is the length at time t, final length also you can say. L naught is length at time 0, or you can call it as initial length, right? R is what? R is growth rate, or in this case, in case of arithmetic growth, you can call R as elongation per unit time, because this R value is telling about growth rate. It's a growth rate. And what kind of growth rate you are measuring? You are measuring elongation per unit time, right? So it's showing you about the rate of elongation. And T is what? T is time, right? So you have to remember only the equation and graph. They can give you question from NCRT, same graph they can place and they will ask you which kind of growth this is, right? Or even they can frame statement that in this kind of growth, uh, only when only one daughter cell divides, another remains meristematic. This kind of statements they can write for this graph given to you, right? So you have to choose that which particular statement is going correct with this graph. Okay. On the other hand, you can see this kind of growth is called geometric growth, right? Just say if you try to plot, okay, initially one cell, then we have two cells, then we got four cells, then we got eight cell, 16, 32, and so on, right? So you can see initially the growth is very, very slow, right? When the growth is very slow, you will call it as a lag phase or a stationary phase, right? Very slow growth, limited cells are there, right? After that you will see there will be rapid increase in the growth, right? From 4 it becomes just double 8, from 8 16, 16 to 32, 32 to 96, right? So many, right? So here uh, here this stage, this particular stage is called exponential stage, right? After this you will see it will not change further. Just like let's suppose I'm writing one number 98, after that it's not changing, right? It's not changing, it is 98 and it's 98 and it's 98 only. Okay, it's not changing, the cells are not dividing further. Then again it is this particular is called, it's a stationary phase. Stationary phase. So we have the first one, lag, exponential, and stationary phase, right? This is the lag phase, exponential phase, and stationary phase. You will see this kind of growth in the size or weight of an organ very easily. right? As per the given time, what is the increase in the size of organ? Initially, when you have seen any fruit getting formed, right, you may have seen it is very tiny, right, because number of cells are limited in that. After some time, you will see it will grow very fast, right? It will grow very fast, right? It's sorry, it's exponential phase. And after some time, you will see there is no growth at all. It will be like that. It does other processes like ripening of that fruit and all will take place. Cell division will stop, right? So at that time, the particular fruit has achieved its stationary phase. Further, it cannot grow, right? And this same thing is represented by W1 equals to W dot E raised to RT, right? So what is this W1? It's a final weight. W naught is the initial weight of that particular organ. E is what? E is the base of natural log. On this also they ask questions many times. What is the meaning of E? E is the base of natural log. What is R? R is growth rate. There is another meaning of R for this. What is that meaning? R means efficiency index, right? For the other equation, right, which is LT equals to L naught RT. For that equation, R means elongation rate per unit time. For this equation, R denotes efficiency index. Efficiency index का meaning क्या है? Ability of a plant to synthesize new plant material, right? Up, just imagine by yourself, if a plant is undergoing growth and development, right? If growth is occurring, then definitely lot of new material needs to be produced in the plant body or not, right? And if I'm saying that the plant is able to synthesize new plant material, means R will increase, growth rate will increase, plant is efficient to show growth. Understood? If I am saying at all that R is very less, okay, value of R is very low, it means plant is not efficiently producing all the materials which are required for growth. That is the meaning of efficiency index or R value, ability of a plant to synthesize a new plant material which is necessary for plant growth, right? So all the things are interconnected and interrelated. You have to understand its meaning, right? This is given in NCRT. You may read NCRT after the lecture. Okay, now please solve this question. The process of growth is maximum during which phase? The process of growth is maximum during, I have told you, you can say exponential phase, right? But in the option there is no exponential phase, right? So one more thing you get from here, exponential phase is also known as, can you guess it? It is log phase. This question they asked in 2020 NEET, so important, right? Exponential phase is also called log phase. Okay, lag phase, log phase, stationary phase. So it is log phase. Dormancy में तो growth is suspended for some time. Senescence तो last stage of development, right? Lag phase में तो it is very slow growth, right? So exponential phase or log phase we have to remember. Typical growth curve in plant is, in if you are considering about entire plant, right? I told you that for elongation you have to consider, or for increase in length you have to consider arithmetic growth. For cell, tissue, organ or entire plant you have to consider which type of growth? You have to consider the second growth, that is called geometric growth. Up, you just tell me geometric growth mein which kind of curve was there? If you see this curve, this graph is sigmoid graph, right? This is called sigmoid curve, okay? Or you can also call it as S shaped curve. What about this curve? This is linear. Understood? So we have to remember here, okay, we have to remember here that we must have here sigmoid type of curve, right? Stair step shaped curve, when you will get something like this, okay? This kind of curve you will get in different seasons, okay? when the plant is experiencing different type of seasons, maybe autumn, okay, winter, okay, then spring, summer, in that you will have this kind of stair step shaped growth curve, right? Otherwise you will be having linear parabolic, you will not have. And this stair shape is also a part of sigmoid only, right? Up now we have another topic, that is two more types of growth are there, that is called absolute growth rate and relative growth rate. What is these two things? If you want to uh, calculate the quantitative amount of growth, like if you want to calculate how much actual growth has happened, then it is called absolute growth, right? If you want to calculate with respect to initial growth how much growth has occurred, that is called relative growth rate. So here you can see that quantitative comparison between the growth of living systems can be achieved by two types of growth, that is measuring absolute and relative. What I have written here, total growth per unit time is called absolute, actual what growth happened. How you will find it? Final growth minus initial growth. What about the second one, that is relative growth? So relative growth में this is the growth per unit time as a percentage of initial growth, matlab as compared to initial growth what percentage growth has happened or occurred, that is called relative growth, right? This you can find by this formula, absolute growth rate divided by initial growth rate into 100, right? So in NCRT they have given us this image, right? Can you calculate the growth rate for both? You can see here, this is, let's suppose, leaf number 1 and this is leaf number 2. First let's calculate the absolute growth here. I will write absolute growth rate as AGR. Okay, in the previous slide also you have seen it is mentioned like this. So absolute growth rate, how will mention? Final minus initial will give us absolute growth rate, which is 5 centimeter square. So this has increased, doubled, right? It was initially only 5 centimeter square, now it has become 10 centimeter square, just double growth, 100% growth happened, right? Here if you consider for this one, so here the initial final size is 55 centimeters, so we will write 55 minus 50, right? So how much? 5 centimeter square growth. तो देखो absolute growth तो same है, absolute growth is same for both the leaf number 1 and leaf number 2. There is no difference in absolute or actual growth. This has also increased in size 5 centimeter square, this has also increased in size 5 centimeter square. But is this growth looking same to you if you consider or compare it with its initial growth or size? No, right? So we have, in this case we have to find relative growth rate. Okay, if we find relative growth rate, what we will write? We will write absolute growth rate, that is 5 centimeter square, divided by initial size, that is 5 centimeter square, into 100, because we have to find the percentage of initial growth, right? So this will be cancelled and your RGR will come as 100%. तो यहाँ पे क्या हुआ है? 100% growth happened, right? Double growth happened, right? But on the other hand, if you try to calculate for this one, relative growth rate, so you will write 5 centimeter square here as well, but the initial size of this leaf was not 5, it was 50, right? So you will write 50 centimeter square here into 100, right? So you know very well what to do. This is 5, okay, this will be cut like this, and if you multiply this, what will be the RGR you will get? Only 10%. So if you compare it from its initial growth, right? So how much percentage of initial growth has occurred here? Only 10%, right? So 10% growth, that is only 5 centimeter actual growth has occurred, right? So because the absolute growth rate are similar, that's why we need to have another value, that is called relative growth rate, that is with respect to its initial size how much percentage growth has taken place. I hope you have understood. They give questions on the basis of this also. Now almost the growth part has been completed, right? With a few things which are left, which you have already studied in plant photosynthesis chapter, I hope, right? So see, there are so many factors which are affecting growth, which are responsible for growth, which I have listed all together here, right? The very first factor is water itself, right? Just imagine, what without water any life can survive on the planet? No, right? So water is a very essential component of living system. It is responsible for cell elongation. I told you that whenever a cell is elongating, okay, it requires a vacuole formation. What is there inside that vacuole? Water is there, right? And dissolved solutes are there, so many other things are there. So main component there is water. So without water, can a cell elongate? No, it's responsible for cell elongation. It is responsible for maintaining turgidity of a cell, right? Cell can maintain its shape because of water only. It will provide pressure to the cell wall, right? It will remain exposed. Like if it is a cell of a leaf, the leaf will be exposed, right? It will be turgid. Maintenance. Okay, turgidity maintenance. Now, medium for enzymatic reactions, the most important thing which you have studied in cell unit of life chapter also, right? If there is no water, metabolic reactions will not take place, right? Because metabolic reactions will occur where they will be requiring a particular, you can say, uh, medium where they have to occur, right? You cannot do any kind of reaction without a medium. So water is acting like a medium where so many reactions can take place. Along with that, there are so many dissolved solutes in this cytoplasm. There are a lot of enzymes, proteins, everything which are necessary for these enzymatic reactions, right? So all these reactions are taking place inside the cytoplasm, and your cytoplasm is made up of water, right? So water is very essential. If water is not there, no growth, right? Oxygen, this is the gas which is very essential for the living organisms, specifically plants, because it is responsible for aerobic respiration, right? So it is required for aerobic respiration. It is responsible for producing cellular energy, ATP, right? Okay. Now, nutrients, different type of nutrients are needed or not, okay, for synthesis of protoplasm. Protoplasm, a living component of the plant, or you can say for acting like a raw material which makes the protoplasm, you require nutrients. Different type of categories are there: macronutrients and micronutrients. Carbon and oxygen, hydrogen, okay, all of these things, nitrogen, okay, all of these things are considered as micronutrients. They are required in very high amount, right? On the other hand, we have micronutrients like zinc and iron, right? There are manganese, like that, many are there, right? These are needed in very, very less amount. In biomolecules, maybe you have studied about them. Then temperature. Temperature of 28 to 30 degrees Celsius is optimum for growth, because on this temperature, or at this temperature, so many metabolic reactions will uh, occur, right, at their optimum speed, right? But temperature which is more than 45 degrees Celsius, it is going to hinder plant growth, right? And process like transpiration, enzyme activity, right, they all will get affected. Enzymes will get denatured, right? Protoplasm can get coagulated, right, because of the protein coagulation. Protein coagulation, as in, if you boil egg, right, you see that protein like it will coagulate, right? It will become that white color albumin protein. Like that, a cell also has lot of protein inside the cytoplasm. So if you are boiling it at not boiling, sorry, if the temperature is exceeding 45 degrees Celsius, right, then definitely there will be coagulation of proteins, and along with that, the protoplasm also will get coagulated. Then no reactions can work properly, right? Enzyme structures will damage. Now, light, very essential for photosynthesis, productivity, pigment synthesis, and for differentiation of tissues, right? If tissue differentiation is not occurring, if plant is not producing any food, right, if pigment itself is not there, then plant can never grow, right? These are some specific things which are given in this topic NCRT. You have, like, in this chapter NCRT, which you have to remember. Gravity, right? Gravity will also affect growth or not. It will determine the direction and orientation and the movement of root, stem, and branches, everything, right? So towards the sorry, away from the gravity, stem will grow. Towards the gravity, root will grow. Gravity is also one of the most important feature for growth, right? Here you have to remember this. I feel that they can ask that for tissue differentiation, do we require light? Because till now they have not asked any question like this. So please be very careful with this thing. Now, which of the following is necessary for growth? Obviously all of these things are necessary for growth, right? Cell enlargement mainly requires what? Cell enlargement requires water, right? We need water for enlarging cell because it is filled in the vacuole. How does water help in enzymatic activities needed for plant growth? Right? What water will do? It will provide medium, right? Water provides ions, or water maintains the pH, or it maintains the temperature. No, it's acting like a medium for the metabolic reactions, right? Okay, now there's one more connecting topic that is called plasticity, which is very easy. This is the topic where you have very questions asked in past also, so don't overlook it. Don't think that we will just skip this topic. It's a one paragraph topic which is very very easy to understand. What is the meaning of plasticity? You all know what is plastic, right? Plastic is something which can be molded into different manners, right? So it's having an ability to change itself, right? Like that, you have to remember the meaning of plasticity is ability to change, right? So here, if a plant is able to change itself, right, how it is changing, you have to remember that plasticity or this ability to of change, it can occur by two methods. Either because of change in the environment of the plant, or because of change in the phase of life of a plant, right? In these two, like by these two ways, a plant can change itself. Now, in NCRT, they have explained this concept plasticity with one example that is called heterophily. Right? Plasticity ka example given in NCRT is heterophily. What is the meaning of heterophily? Heterophily means formation of two different type of leaf on one single plant. Right? You have one plant, but that one plant is producing two different types of leaf. That is called heterophily. It's an example of plasticity, ability of change. Right? Ability to change. Right? Now, first thing is based on changing environment. You have to remember this example. There is one example called buttercup. Its scientific name is ranunculus, but it is not given in NCRT. Okay, this buttercup plant or this ranunculus plant, it will start producing two different kind of leaves because of change in environment, right? Whereas these three, cotton, larkspur and coriander, they will be producing two different type of leaf because of change in the phase of life. So cotton, larkspur and coriander, you have to remember. Up here I have one image which is given in NCRT. Dekho, what is the meaning of change in environment? Right, the very first type of example, that was buttercup. That buttercup is a plant which is like which is present half in the water and half out of water, right? So let's suppose this is the water part, right? The part of plant which is under the water will produce dissected leaves like this, and the part which is above the water will produce leaves with broad lamina, right? In case of this buttercup, right, in buttercup you will be having this condition, right? So the part which is water habitat, which is present in water habitat, it's showing us this leaf, and the part which is terrestrial, or in this case we can consider it is not in water, right? It is let's suppose aerial. Is out of water. Okay, that leaf is different, right? What is the reason behind this? This is having broad leaf, and here you have dissected lamina. Okay, this happens because if a part of the plant is under the water, it will receive very less amount of sunlight, right? This part receives sunlight which is less. Okay, this will receive more sunlight. So if this leaf will receive more sunlight, definitely it should have a broad leaf lamina. so that it should have lot of photosynthetic pigment there, right? If we have a broader leaf lamina, there will be more pigment, and if there is more pigment, then there will be more photosynthesis, right? Whereas on the other side, you can see the leaf which is under the water, which is present in the aquatic condition, or you can say in the water habitat, you can see the leaves are not broad, right? They are having very needle like condition, right? So because the exposure of the light is very less, right? So there is no need of wasting energy in making the lamina very broad, right? So this is what, this is a plastic behavior. This is example of two different types of leaf heterophyly, right, which is occurring because of the change in the environment, right? Okay, buttercup, you have to remember this. You have seen coriander, right? You all might have seen coriander plant. Coriander, maybe you have certain leaves which are very new leaves, and then there are certain leaves which are old leaves. Okay, if not, then please see coriander today itself, right? It will be there in your fridge maybe. When you will see coriander, you will see the mature leaves, or you can say the old leaves. They have broad lamina, right? And the new leaves, juvenile or young leaves, right? What condition they have? They have dissected lamina, right? So in this coriander plant, okay, like this one more plant is there, that is called larkspur. Okay, okay, coriander, larkspur. Okay, and one more is there, right? That is cotton. In these three plants, you will be able to see that due to change in the life cycle stage, that is juvenile and mature, there will be a change in the type of leaf, right? Young leaves will be dissected, okay? Mature leaf will be having broader lamina, right? Same thing is mentioned here, juvenile, adult, okay? Here they have not drawn it very correctly, but you have to remember that where lamina is bigger, okay, that is a adult leaf. Where the lamina is very reduced, that is a juvenile leaf, right? And here terrestrial and water. Maybe they can give you this and they can ask you what are the exam, like this is example of what, right? So you have to remember, in image A is given for larkspur, which belongs to change in the phase of life cycle, right? Then there is one figure which is given in NCRT, that is this figure, where you can see they have discussed about the sequence of the developmental process in plant cell, right? So this figure is talking about the development in an individual plant cell. Okay, although this diagram could have been a very better diagram, but NCRT has given it, so we have to take it as it is. You have to consider they have started here as a meristematic cell, right? So there is a cell which is going to divide here, right? It's a meristematic cell. When a cell has to divide, what is the condition? It has to give rise to two daughter cells, right? So let's suppose it has given rise to one daughter cell and another daughter cell, right? One daughter cell will remain meristematic. So after plasmatic growth, right, after somewhat increasing in the size, not completely fully increasing, but yes, as per parent it will increase. After achieving some plasmatic growth, you can see the cell is reversing to divide again, right? So this is talking about the fate of this one cell, which is going to again become meristematic. It will divide, right, after completing some of the plasmatic growth, matlab it will increase in the size. Then the second daughter cell, you can see, as soon as it will divide, after division the plasmatic growth will begin, matlab the protoplasm will increase in size. What is the meaning of plasmatic growth? Increase in protoplasm, Understood? You have studied after the division. This is cell division only, right? You can see here, after the division, the daughter cells must undergo elongation. So they are entering in the elongation. They have started plasmatic growth, in which one will be again selected for remaining meristematic. Another has to become permanent. So I'm writing here, this is meristematic. This one is permanent cell. The one which has to change into permanent cell, it will start doing plasmatic growth, right? As it is start doing like as it is doing plasmatic growth, you will say it is expanding, it is under expansion, or you can say it is elongating, it's under elongation, right? As elongation is going to complete, before that you can see differentiation will begin, right? So the two arrows which are separating here, they want to denote this only, that it's not like one step is completed. Don't think that okay, first cell will divide, cell division achieved, yes, then what will happen? Cell elongation will start, right? But elongation is not completed. Much before that, see, elongation is not completed, but the cell started to get differentiated. So it is occurring in step by step manner, but it is not like one entire step is over, then another will start. No, there is overlap in between. Are you able to understand? This is the thing which we have to take from this diagram. That is the way they have drawn it, right? As the meristematic cell has to become permanent cell, it has entered into plasmatic growth, where elongation of the cell will occur, vacuolation will take place, cell wall synthesis will occur, right? Cell will expand in size. As the cell is expanding in size, you will see some changes will start occurring in its protoplasm, right? Or the cell is going to differentiate. And I told you, in differentiation there will be structural and physiological changes. Remember, I told you there will be changes in the structure and physiology of the cell, so that will occur, right? So you can see differentiation is occurring, cell is maturing, and finally the cell has become a mature cell. What is the another name you have studied for this? It is a permanent cell. Now the cell is specified to perform a specific function. Now the cell will not divide, it has lost the division capacity. Now the cell will enter in the stage of, you can say, a stage which will lead to death, that is called senescence stage, right? So you have to remember, this is the pathway of the cell which has to become mature and ultimately it will die, and this is the pathway of a cell which has to keep on dividing, right? So there are two daughter cells which are formed from one meristematic cell, and both cells ka developmental weight they have given in this diagram. Okay, now can you please try to solve this question, right? Please solve this question. I hope you are able to solve, right? You can see here they have given first one, they have not written. So tell me, what is the name of the first thing? What we should write here? First, they have written there as meristematic cell. So what you are going to choose here, where they have mentioned meristematic cell? And it is very easy question, because only by getting that you can get the answer, right? So here we have meristematic cell. Then what is second? Second may you have to remember, expansion is occurring because of what? Plasmatic growth, right? This above arrow is denoting plasmatic growth, right? Because of here differentiation also started, right? Actually they should have, okay, made it little bit better. Yahan pe what we should write? We should write here maturation, right? Differentiation is occurring in maturation phase. Cell will finally mature and it will undergo senescence, right? So we got the answer, it is option number 2, that is option B. Okay, easy question. They have not given any question with respect to this diagram, so there is a possibility that they can give a question. You have to be very careful, right? Now, can you solve this question? In arithmetic growth, what happens to the cells other than daughter cells? Okay, arithmetic growth is the first type of growth, right? Where one meristematic cell divides, it gives rise to one meristematic cell, one permanent cell. This will again divide, right? It will give rise to one meristematic cell, one permanent cell, and this permanent cell I will write like this, right? So arithmetic growth, what happens to the cells other than daughter cell? Okay, so these are the daughter cells, these are the daughter cells, right? They are talking about this cell. Again, let's suppose I am writing for one more generation. So this will be meristematic cell, this will be permanent cell, okay, and then I will be having two daughter cell, right? So what will happen to the daughter cell? They undergo maturation, they undergo differentiation, they will undergo continuous cell division, or both A and B. You got the answer? I have written that these are permanent cells, so they have to enter in maturation phase, right? They have to undergo differentiation in maturation phase. So option D should be the correct answer. Are you able to get the point? That here meristematic cell will keep on dividing, but the fate of permanent cell is that they have to change themselves, they have to differentiate, and they have to become mature, right? A sigmoid or S curve is characteristic of, right? So sigmoid or S curve is there in which growth? Geometric or arithmetic? It is geometric, right? So where you are going to find it? Living organism growing in a natural environment, typically all cells, tissues and organs of a plant, and root elongation at a constant rate. So this we cannot choose, we know very well this is an example of arithmetic growth, right? We can choose this, and we can combine this option with this also, right? Because you see, if you get any time a graph like this, right, S-shaped graph, this was also there in one of the growth which you have studied, that was logistic growth. Remember, in organism and population chapter, there is a name which you have studied which is called logistic growth, right? Exponential growth and logistic growth. And logistic growth has a concept which is called carrying capacity, right? This is the nature's environment resistance, okay, the limited supply of natural resources which are available for any kind of population which is growing, right? There is always a block from nature side, right? So yes, living organism which is growing in a natural environment, it can have nutrient supply limitation, it can have limited amount of carbon dioxide, right? In photosynthesis chapter you have studied, right? Ma'am has taught you, right? You have studied, there can be any predator also, there can be any kind of parasitic disease also, right? So so many environmental resistance are working together so that they can limit the growth of a particular organism, right? So that is what, in a natural environment, if any particular plant is growing, definitely it will be affected by this thing, right? So yes, we have to choose A and B, okay? If not, you open organism and population chapter, there is a topic exponential growth, please read it, right? You will be able to understand why this is there. Although I have told you that carrying capacity or limited nutrient supply along with a lot of resistance from the nature are there, which are working all together to, you can say, make the growth of living organisms constant, right, after some amount of time. Okay, fine. Now this chapter is having the same thing which is given anatomy of flowering plants also, that is differentiation, redifferentiation and dedifferentiation, right? So we have to remember these processes because this is very very mandatory for understanding the plant growth and development, right? So what is the meaning of differentiation? You can read this, I have mentioned it in every possible way from NCERT, that it's the phenomena by which the cells derived from root apical meristem and shoot apical meristem, they will undergo, right, they will undergo, they undergoes permanent changes in their structure, biochemistry, size, physiology of cell wall and protoplasm, right? What all changes will be there? There will be permanent changes in the structure of a cell, biochemistry of a cell, physiology of the cell, and cell wall and protoplasm, enabling the cell to perform a specific function, right? By these changes only the cell is going to perform a specific function. And I have given the examples given in NCERT. They have given the differentiation of trachyria element and chlorenchyma. Okay, what differentiation they have given? For converting into trachete or maybe vessel, which is trachary element, the cells must elongate, they must lose their protoplasm, they should have a strong elastic lignocellulosic secondary cell wall, right? Primary cell wall is there in meristematic cell. Ab dekho, you have to understand it like this: how a primary meristem ka daughter cell, which was having the features of meristematic cell, have to change into a permanent cell or mature cell. Let's suppose this permanent cell which we want to make is a cell of a xylem. Okay, trachary element means either it can be a trachete or it can be a vessel. Both are dead cells at maturity. Both needs to conduct water. What is the purpose of meristematic cell? Meristematic cell ka purpose is that that cell has to keep on dividing. So for that, what feature should it have? It should have dense protoplasm, thin cell wall. It should have a big nucleus, right? It should have ATP synthesis. Lot of anabolic reactions are occurring. These all things are there in a cell which is ready to divide. You want to make a cell which is going to transport water, right? Which needs to conduct water with the like inside the xylem, right? Or you can say it has to conduct water and has to become a part of xylem tissue. Uske liye what are the things which needs to be changed, or what kind of differentiation is needed, right? What kind of change in the structure, what kind of biochemical change in the cell, what kind of size change, what kind of physiological activity changes, what kind of cell wall and protoplasm changes have to occur, so that I can make that meristematic cell daughter cell as trachete or vessel cell. Same I have written here. The cell must elongate. First of all, the cell size should elongate, right? Then the cell should start losing its living component, that is protoplasm, right? So that space can be created for the conduction of water, right? The cell should change its cell wall biochemistry, right? The cell wall normally has cellulose, but that cell wall must be changed. Cellulose must be replaced by lignin plus cellulose, so it should be lignocellulosic, right? Under this much water tension, the cell should not break or damage, so it should be strong plus elastic, right? These kind of changes should take place. Techids, they do not collapse under high water tension also, and they can carry water to long distances. This is their function. This function this trachid is able to do because of the changes which it has acquired during differentiation process or not. Now, can this cell do any other work? No. It has become specified for this function only because of these kind of changes. Are you able to understand? Right? The second example which explains differentiation is chlorenchyma, right? Which is a photosynthetic tissue, chlorenchyma. The example for this is mesophyll cell, right? So let's suppose I have a meristematic cell with some few characters I have quoted. Vacuole is not there, cell wall is a primary cell wall, cellulose wala cell wall, it's having dense protoplasm, big nucleus, right? This cell I want to differentiate, change into क्लोरेनकाइमा का cell that you can consider as a mesophyll cell. इसका example you can take as mesophyll. Okay, here trachid and vessel, here mesophyll cell. So what kind of changes are necessary? If you want to convert meristematic cell into mesophyll cell, the chloroplast synthesis in leaf should begin, right? Chloroplast formation in leaves should begin. Okay, and along with that, okay, in leaves I should have written here, along with that there must be thin cell wall. Okay, there must not be lignocellulosic cell wall must be thin, and then only it can be allowed to perform a specialized function that is called photosynthesis. So you have to remember this, because plants are able to differentiate throughout their life, right? Differentiation process is never going to end in the plant body. If the cells are dividing, definitely the cells have to perform their roles. For performing their specific role, they have to undergo differentiation. Because differentiation occurs throughout the life of plant, that's why we can consider differentiation is also open in plant, right? We cannot do this. right? From a single meristematic cell we cannot make different kind of cell depending upon different change, right? We can make only one kind of cell from that progenitor cell, right? That also is very limited, only in our bone marrow that possibility is there, otherwise it is not there. But here in plants they are open for differentiation just like growth, right? What is de-differentiation then? One process to you understood, that from meristematic cell if you want to make any kind of cell in the plant body then it will be formed by differentiation. But sometimes you will see that sometimes there is one meristematic cell which is not present at the tip. There are some meristems which are present at the you can say at the lateral part, or you can say in the center of the plant body, right? That meristem is called vascular cambium and cork cambium. So this is what, this is secondary meristem which is going to develop later in the life of plant, not there from the beginning, right? If you want to develop secondary meristem you can do that by the process of de-differentiation. So the cells they lose their division capacity or the ability to uh, divide when they are differentiating. Here there is no division capacity. One trachea cannot divide to make another trachea. One mesophyll cannot divide to make another mesophyll. One epidermal cell cannot divide to make another epidermal cell. It is not possible, right? Okay. So they have lost division capacity. Meristematic cells, primary meristematic cells में division capacity was present. What is primary meristematic cell? Root apical meristem and shoot apical meristem. Division capacity present there. After differentiation, whatever daughter cells we have made, sorry, whatever cells we have made, diffusion capacity is lost. Now, some of these differentiated cells, if we de-differentiate them, then again they will regain the again they will regain the division capacity. This is called de-differentiation, right? So de-differentiation is there to make secondary meristem. Always remember, de-differentiation is only is done in plant body for making secondary meristem, right? What are those secondary meristems? Secondary meristems are cork cambium and vascular cambium. I will be discussing about anatomy of flowering plant chapter. There I will give a brief idea about this, right? Right now you have to memorize and learn and revise these points only, that there are certain primary permanent cells like medullary rekha cells and cortex cells. These cells only will de-differentiate and they will form interfascicular cambium, which is a part of vascular cambium, and this cell will form cork cambium, right, which is also a type of secondary meristem, right? So you have to remember de-differentiation. Redifferentiation, whatever these secondary meristems are going to make, right, that is called redifferentiation. The process by which redifferentiated cells again lose their division capacity, okay, and they become permanent, that is called redifferentiation. Only four tissues are redifferentiated tissues given in NCRT, which is now not given, I think it's given in old NCRT, okay, but you may remember it, maybe they can ask a question. Secondary phloem, secondary xylem, secondary cortex and cork cells, all of these structures, all of these tissues are made by redifferentiation, okay. So this is there, it completes most of the part of this chapter, okay. If you want I can tell you one more thing about this differentiation de-differentiation concept, right? But I think I should hold it for anatomy of flowering plants because already it will become very lengthy for you. If you can remember that for making secondary meristem which process is required, de-differentiation. For making the secondary permanent tissue which process is required, that is called redifferentiation, right? And for making all the cells from primary meristem, what is required? Differentiation. If you can remember this, it's enough for you to do questions. The cells derived from root apical meristem and shoot apical meristem and cambium differentiate and mature to perform specific functions. Okay, this act leading to the maturation is termed as, so we can do here differentiation. Okay, here the problem is cambium. Cambium should not be written here. Okay, cambium should not be written here. If I'm talking about cambium, okay, and this cambium can be of two type, vascular cambium or cork cambium. Whenever any kind of tissue is formed from this cambium, Okay, or I can write here secondary meristem for cambium. In some other way we can take this up, because there is one more cambium that is called intrafascicular cambium. Okay, if intrafascicular cambium is dividing, that is primary meristem in origin. Before I'm writing it, you will be able to understand. Okay, whether it is root apical meristem or shoot apical meristem, both are primary in nature, or intrafascicular cambium, this is also primary in nature. Okay, if this divides, it will do differentiation always, right? And it will form the particular structures, right? They will form certain cells, okay, which are permanent cells. This will also form permanent cells, but because it is secondary meristem, all the permanent cell will be called secondary permanent. That's why we have written secondary xylem, secondary phloem, secondary cortex, right? So this will be formed from redifferentiation, because this is second time you are doing differentiation in the plant body, right? Up, if I want to consider this question correct, right? Let's suppose I want to consider this I want to say that yes, this question is correct. So what I have to do? Okay, what I have to do? I have to consider that I am talking about which cambium, intrafascicular cambium. Then though I can take differentiation here, right? So I'll choose differentiation here. Okay, then also I think we can exclude this part here. Okay, in anatomy of flowering plants, when we'll do revision, I'll give you a very good idea about these questions, right? What changes would occur in a cell to form a tracheary element, right? If you want to make tracheary element, it should lose all the protoplasm, right? It should lose all the protoplasm, sorry. It should develop a strong elastic lignocellulose secondary cell wall. It should never develop vacuole, right? No need of vacuole. Vacuole is a living cell, it's a dead cell, right? Redifferentiation is a process in which what is going to happen? Cells lose capacity to divide and get mature. Cells divide to perform specific functions. Cells undergo senescence, or cells regain the capacity to divide. So we can say that when the cell is doing redifferentiation, cell is regaining the capacity to divide, right? The redifferentiation means cell has regained the capacity to divide. Cells regain the capacity to divide. Okay. Redifferentiated tissue will lose the you can say capacity to divide. Okay, formation of interfascicular cambium from fully developed parenchyma cell is example of. So I mentioned intra, that is primary, inter is secondary. Tell me, secondary meristems are developed by which type of thing? Secondary meristem, I told you, if you want to make secondary meristem, right, you have to do de-differentiation. Okay, fine. So one part of our chapter is completed. Now we have to complete the next part of the chapter, that is the growth hormones, right? When you study, I'll give you one brief idea about this. For plants growth, if you consider plants growth, for plants growth and development, okay, there are so many things which are required. So many factors are going to affect this. Okay, so the factors which are going to affect it, okay, they can be external factors, sorry, they can be intercellular factors, or they can be intracellular factor. Okay. Intercellular factor we have to study here. Intercellular factor में what we have to study? We have to study these PGRs. In between the cell, these hormones are synthesized, right? These hormones are going to affect the development of plant, right? Intracellular factor may we have to remember these are genetic factors, okay, which are present inside nucleus. So there's a chapter called Principles of Inheritance and Variation. About genetic factors we are going to read there. जैसे कि we write ना, if plant is tall, it will be having this uh, allelic pair. If it is tall, it should have this kind of allelic pair. If it is dwarf, it should be small t, right? So this we have to study in Principles of Inheritance chapter. This we have to study in this chapter. And these are only plant factors which I'm talking about, or cellular factor. There are extrinsic factors also which are there, which you have studied in photosynthesis chapter. So I have not written it here, right? Photosynthesis me you have nicely studied what are the extrinsic factors which are going to affect the plant. Okay, so here what are these PGRs or plant growth regulators? Very easy topic, and lot of questions you will see they have asked from these topics, right? Every year this chapter will give you 3 to 4 questions, right? Very basic chapter, very easy chapter. Understanding is very easy, but you have to memorize so many things, right? So please memorize it. Please take up those free marks of 4 marks in each and every question which this chapter is going to pay you, right? So plant growth regulator PGR can be considered as these are the hormones which plants will synthesize in very low concentration, and they will be having a very effective role on overall growth and development of the plant, right? Now basically we can divide all the plant growth hormones under 2 categories. First, you can see here I have written as the growth promoting hormones, right? Or the PGRs which are involved in growth promoting activities. And what are these growth promoting activities? Cell division, cell enlargement, right? Pattern formation, right? The plant pattern, how the body is getting formed, right? Where should be the stem, where should be the root? So pattern formation, tropic growth, right? Movement of the plant, fruiting, right? Seed formation, right? All of these are considered as growth promoting activities. These growth promoting activities are actually promoted by, or they are regulated by certain hormones like auxin, cytokinin and gibberellic acid, right? These 3 names you have to remember. These are considered as plant growth promoters. Plant growth inhibitors, here you have to remember, here we have growth inhibiting roles, right, which will be done by certain plant growth regulators like abscisic acid. Together we will say that these are the group of these are the hormones which are involved in plant growth inhibiting activity, right? So abscisic acid is the only one which is involved in such kind of activities like dormancy, abscission, right? So anyways, this is good for the plant. If the plant is dormant, the plant will get enough time to develop its seed, right? If the seed is under dormant conditions, it will get time for maturation, it will get time for development, and when the correct conditions are there, the seed will germinate. So it's not bad for the plant, right? It's only helping the plant, but there is no division, no enlargement of the cell, no synthesis of any new thing. That's why it is clubbed as plant growth inhibitor. Then there is the fifth hormone that is called ethylene. Okay, ethylene is a only gaseous hormone, and some of its role are inhibitory in nature, some of the roles are promoting in nature. That's why it is considered as uh, you can say a transitional type of hormone, where some both type of roles you are going to see, right? Now see, they will give a question on the basis of this also, that which of the PGRs are promoters. So you know very well, auxin, gibberellic acid and cytokinin together can be considered as plant growth promoters, right? Now before entering to this, I'll tell you about some more things, okay? जैसे कि total you have auxin, 5 hormones are there, right? You have auxin, you have gibberellic acid, cytokinin is there, abscisic acid and ethylene is there, right? अब इसका precursor you have to remember. Okay, what is the precursor of, okay, these are the hormones I'm writing here, and these are the precursors I have to write here. Okay. For auxin, you have to remember, iska precursor is, okay, tryptophan. This is not given in NCERT. What is tryptophan? Tryptophan is one amino acid, right? So tryptophan is having one aromatic ring that is called indole ring, right? And maybe you know that its natural form is indole acetic acid or indole butyric acid, right? This is the natural form of this oxide. It is derived from this indole containing compound, that is tryptophan, right? Gibberellic acid is derived from terpenes, okay? And the first isolated gibberellic acid was gibberellic acid 3. Naturally, it is present as this only. Cytokinin is adenine derivative. Okay. Adenine is what? It's a nitrogen base. So if any question is there which will ask you that which among the following hormones are basic in nature, so you have to write cytokinin because it's derived from adenine, which is a base, right? Acidic, this is acidic, this is acidic, abscisic acid is also acidic, right? Abscisic acid, so they have not given that it is derived from what, okay? So but it's some carotenoids are there through which it is derived. Then ethylene, you have to remember there is one amino acid which is called methionine, so it is methionine derivative, right? And it is the only hormone which is gaseous in nature, okay? And it is mentioned as C2H2, double bond C2H2, or CH2, sorry. Okay, so you have to do this, right? You have to remember that they are derived from where. Now let's learn about the discovery of the very first hormone which was discovered, that is auxin. Auxin actually was worked extensively by many scientists, and Charles Darwin, right, and his son Francis Darwin, right, these are the two scientists who were studying one particular phenomena that is called phototropism, right? When you apply a unilateral beam of light at the tip of a plant, you will observe a bending of the tip towards the plant, right? So this bending occurs due to light stimulus. This is called phototropism, right? Bending of any particular part of plant towards the light, right? So they were studying this phototropism, how bending takes place in which plant? In canary grass, right? They were studying this phototropism in canary grass, and there they saw that bending is getting like bending is observed when the unilateral light is illuminated to the tip of canary grass, right? Now, but they did not understood what is actually responsible for that, okay? Then FW went, okay, he isolated auxin, okay? FW went was the scientist who isolated auxin from the tip of, okay, oat seedling, or you can say it avena. Its plant name is Avena sativa, okay? From the tip of this oat seedling or Avena sativa, they have isolated, like FW went have isolated auxin for the first time, right? Then the first hormone to be isolated from human urine, right? From human urine, this hormone was isolated for the first time. You must be thinking what this hormone is doing in human urine, right? So you have to remember that it is formed by the metabolism of tryptophan. Tryptophan amino acid is there in humans also, right? So indole acetic acid or indole butyric acid type of compound was found in human urine also, because there is no use of this compound in human body, so it has been excreted out, passed out in the urine, right? So we isolated it and we found that this compound is nothing but a kind of auxin, right? Now IA and IB are isolated from plants, so can we say IAA and IBA, these are natural auxins? Sometimes they will give a question, right? This we have isolated from plant, so these are natural auxins, right? What about this two, naphthalene acetic acid and 2,4-dichlorophenolic acetic acid? These are actually artificial These are artificially synthesized oxygens which are synthesized in labs so that we can use them and we can apply them for many particular functions which organs they do, right? So you have to remember all these things. Okay, who was the person who was studying phototropism in the which plant, canary grass plant, right? Then who was the scientist who is responsible for synthesizing oxygen from oat seedling, that was FW Wendt, right? Now early phototropic studies have explained about this. So what they have mentioned here, you can see in the first one, when they started illuminating the tip with the unilateral, right, they observed a bending, right? So they thought that okay, something is getting formed here, that they have called as transmittable compound. Some transmittable compound has formed here and it is going away from the tip and it is allowing bending. To confirm it further, what they have done, they have cut or decapitated the tip of the uh, plant, right? So if we remove the tip or we decapitation, it is called decapitation, removal of tip. Okay, if we do decapitation, what will happen? There is no tip. Now if you illuminate it with the uh, uh, light, what will happen? There will be no bending. So what we understood, for bending, the stimulus must be perceived at the tip. Tip is necessary for bending, right? So that chemical will not be formed if the tip is removed. Then further what they have done, they have covered the tip with one opaque cap, right? This opaque cap was not allowing any light to pass through it, and as a result, again the tip which was intact, which was having one covering like tin cuff foil, you just wrap it, right? So it's opaque only, there will be no light signal perseverance on the tip. What will happen? There will be no bending, right? Then what they have done, they have covered the tip with a translucent or transparent type of cap, right? जैसे कि प्लास्टिक से कवर कर दो और you cover it with some other material which is transparent. What will happen? The light will be perceived at the tip and you will see bending is observed. So by this series of experiments, we understood that tip is necessary for taking the stimulus. Okay, light is necessary for this stimulus to be perceived at the tip, and by receiving the stimulus there is some transmittable compound which is getting produced at the tip, then it is moving away from the tip and allowing bending. Actually what happens, this auxin will be responsible for elongating the cells in this side, right? And this is called differential elongation. If cells are elongating at this side and not elongating at this side, you will observe a bending or not, right? So that was not found as per NCRT. That is what happens actually, which you should know, right? Now these are the important things which you have to remember. This auxin will do what in the plant body? These are the physiological effects of auxin. Along with that, what are the applications which we can get using this auxin hormone in our plants, right? So see, first thing, auxin is that particular hormone which initiates rooting in the stem cutting. If you have any kind of stem and you want to grow, or vegetatively you want to propagate it, what you can do, you can cut the woody stem and you can dip it in auxin solution or maybe auxin powder, and you will see that there will be rooting or root formation which will be initiated in these stem cuttings, right? Then in pineapples, auxin application has, sorry, in pineapple actually you will see that auxin is promoting flowering, right? So flowering in the pineapple is promoted by auxin. As you have seen, auxin is a plant growth promoter, so flowering is promoted by auxin. You have to learn this, right? It helps to prevent fruit drop when the fruit and even leaf also, when the fruit and leaf are very young, at that time they need to develop and grow, right? So in that case, the auxin is going to prevent the leaf or fruit drop. That is called abscission. Abscission means, if let's suppose a parent plant is there and on that parent plant there are so many leaves, so many fruits are attached. So if the leaf and fruit is getting detached from the parent and falling down, that is called abscission, right? So auxin is seen to prevent abscission in the young fruits and young leaf, right? So it will prevent the leaf drop, fruit drop, and it will promote abscission, right? It will allow or promote the leaf drop and fruit drop in the old mature leaves, right? Because now the old leaves, mature leaves, or mature fruits, they are completely developed. Further, they don't need to grow, so it must be removed from the plant to save energy. Auxin has the dual roles here. When it is growing stage, young stage, it will prevent abscission. When the same will become old, mature, it will promote abscission. You have to remember, they will reverse the statement and they will give you, right? Now, responsible for apical dominance. You all know apical dominance, right? If the tip is intact, definitely it will be producing auxin. Auxin is an inhibitor of growth of lateral shoots. That's why you have seen that in during the uh, when you visit on any hilly areas near the tea plantations, what you have observed, the people with big scissors, they will be pinching off the tips, right? Because in the tea plants you require more leaves, and more leaves will be produced when you have more branches, right? So more branches will be grown, more branches will be visible, okay, only when the tip is removed, so that that dominance or inhibitory effect of auxin is removed, right? Once the auxin is removed, then the lateral buds, they will start growing, because cytokinin will start working there, right? Always remember, auxin is inhibitor of cytokinin. It has a negative role. Cytokinin is responsible for lateral shoot formation, right? So if you want more bushy plant, you need more cytokinin. If you need more cytokinin, you have to remove auxin. Where is auxin present? At the tip, right? So this you have to remember. Now, auxin has seen to induce parthenocarpy in tomato, right? Tomato, you can make seedless tomato using auxin. Then these are the applications, right? If we want to make weed free lawn, right, then we have to kill all the dicot weeds, right? Okay, if you want to prepare weed free lawns may, what is the thing which require lush green grass, right? Grass is a monocot, right? And broad leafed plant, right? Dicot plant, right? Dicot plant, mostly small dicot plants will grow there in the lawn like weeds. So we want to kill those dicot weeds. For that we can use 2,4-D, 2,4 dichloro phenoxyacetic acid. What this will do, it's like an artificial auxin. It will selectively kill only dicot weeds, it will not kill the monocot plants, right? So we have to use this for building uh, weed free lawns. Then another thing we have to remember is, auxin also controls xylem differentiation, right? Different types of cells in the xylem tissue, they are formed, they are differentiated because of auxin amount should be there, right? And it also helps in cell division, right? If cambium activity is going on somewhere, meristematic activity is going on somewhere, cell division process is occurring, then auxin must be there, right? Auxin promotes cell division, xylem differentiation. These are some of the physiological effects of auxin which we have to remember, right? This is the ncrt image of apical dominance which you have to consider. Here you can see the tip is intact, so auxin will be produced here. If the tip is intact, can you see these are the lateral buds? These lateral buds are not able to grow, right? Lateral buds are not growing because tip is intact, auxin concentration is very high. So if auxin is there, it will definitely inhibit the cytokinin activity of let present near the lateral bud. But if you pinch off this tip, if you decapitate or cut it, can you see there's a growth in the branches, right? The lateral buds are growing into branches, right? So this is because cytokinesis is not now occurring, cytokinin is ready to function, its inhibitor has been removed, right? So you have to remember, a plant with apical bud intact, a plant with apical bud removed, right? And note the growth of lateral buds into branches after decapitation. Decapitation means removal of the tip. Now can you solve this question? Please solve. This is very easy question. Auxin was isolated from tips of polyptyles of which plant? Hmm? Isolated they are writing, not phototropism. Okay, it is isolated from oat seedling, right? By FW Went. Okay, they always ask question on this. Bending of stem or coleoptile or shoot towards light if potted plant is placed near a window is due to, okay, greater light availability at the tip. Are you able to understand? Right? Because this bending is observed because of only light, right? More oxygen content on shaded side. Okay, this so we can, okay, let us see. Greater oxygen availability at the tip. No. More oxygen content on shaded side. Availability of necessary warmth at the tip. Okay, this option should not be correct. Can you guess why? Okay, bending of stem or shoot towards light is, sorry, if light, if towards light if potted plant is placed near a window. Okay, अब देखो यहाँ पे क्या है. There is a potted plant here and it's placed near a window. So light will fall like this from one direction only, right? And bending is observed toward the light. अब this bending is observed, let's suppose light is coming like this, because oxygen is getting formed here. This is the illuminated side and this is the dark side, right? And I told you after getting formed the oxygen will go towards the dark side, right? And then what it will do, it will allow elongation of the cell like this. Here the cell are of normal size, but can you see here the cells have elongated, and as a result the bending has been done, right? So here what we have to do, more oxygen content on the shaded side, right? This is not given in NCRT, okay, but you have to remember more oxygen is there on the shaded side, it will actually get transmitted towards this side after getting developed here at the tip, right? And why we can't choose this, because you know how I got the answer, it is mentioned more light availability at the tip. No, it is from one side, right? We are eliminating the tip from one side, unilateral light, right? So we have to remember this is the most correct answer. The phenomena which is influenced by oxide and also played a major role in its discovery, that is phototropism only or not. Bending occurred because of oxygen and it lead to the discovery of oxygen, right? Apical dominance is caused by oxygen in shoot tip, right? Tip pe there is oxygen, it is not allowing the growth of lateral buds. Then, which of the following PGR promotes abscission of older and mature leaf? So that is also oxide only or not? It will prevent the, sorry, it will prevent the, uh, you can say leaf and fruit fall in the young leaf, sorry, in the young condition, and will promote the abscission of older mature leaf and fruit in the old condition, right? Now the second one, that is gibberellins, right? So gibberellins or gibberellic acid, right? It's the second category of hormone which we have to study. This is also plant growth promoter only. This discovery, acha, one more thing I will tell you. All the plant hormones are discovered accidentally, right? They were not actually discovering the hormones. People were studying some other phenomena and they got to know this phenomena is occurring because of this hormone. जैसे कि phototropism was being studied and later it was found that this phototropic movements are because of auxin hormone. Like that, gibberellic acid was discovered because there was one particular scientist, right? E. Kurosawa. E. Kurosawa was studying one disease in rice seedling that is called foolish seedling disease, right? Foolish seedling disease or Bacchane disease, right? Then what he found in this disease, that rice seedling is getting elongated very fastly and it is not able to survive for a long time. As soon as germination is occurring, rice seedling is abnormally increasing in the length and it is falling down very quickly, right? So that disease is called foolish seedling disease. Later some more scientists and co-workers of E. Kurosawa have isolated one bioactive chemical from this seedling. This chemical was considered as uh, gibberellic acid, right? Because it was getting formed from a fungal pathogen, Gibberella fujikuri, right? So actually this disease in the rice seedling was because of infection of that seedling by one fungus that is called Gibberella fujikuri, and this Gibberella fujikuri was producing one bioactive compound that is called gibberellic acid, and because of gibberellic acid's action there was elongation in the cells, and as a result the rice seedling was growing abnormally, right? So gibberellic acid 3 is the first gibberellic acid which was discovered and studied extensively, and all the gibberellic acids are acidic in nature, right? These are the things you have to remember. Now the most important question will be asked, what is the role of gibberellic acid? So one thing you have to remember, wherever there is increase in the length, elongation in the length, right, except a few cases, you will see there will be gibberellic acid. Mostly it has to be responsible for elongating the stem. Now here you can see increased length of grape stalk, right? Grape stalk ka length is increased because of application of gibberellic acid. Definitely if any grape stalk is there which is having more length, right, it will produce more grapes or not, right? It will produce more, right? So this increase in the length of the stalk of grape is because of gibberellic acid. It will also be responsible for increasing the uh, length of apple, right, as well as improving the shape of the apple. So you have to remember it causes fruits like apple to elongate. Also it will be responsible for improving the shape of that apple. This is the particular hormone which will be delaying the senescence period, right? It will delay the senescence period, and as a result what will happen, there will be the extension in the market period, right? So you have to remember here, extends market period for fruits. Okay, what's the meaning of this? Meaning is that, let's suppose if you are a farmer and you have applied gibberellic acid to one batch of your crop, what will happen when, like, let's suppose now there is a season for let's suppose mango, right? So mango season is there. So whenever mango is mature, it will fall down or drop down from the paid plant, and you have to sell it in the market. So when there is season, that fruit is very cheap, right? It will be purchased in very less amount of money. But let's suppose after 4 months, if I have applied one batch with gibberellic acid, and it is going to extend the you can say maturation period, and it is going to delay the senescence, then my particular fruit will ripen after 4 months, right? So by that time this growing period will be over, season will be over. Then that particular farmer can come up, right, with mangoes in the market where no one else is selling mangoes. So it is extending the market period of the fruit, right? And as a result we can produce fruits for you can say more money, right? But you have to remember only this, what is the physiological aspect, right? You have to remember that it is delaying the senescence period, and it is because of this we can extend the market period of fruit, because fruit can be residing for a longer duration on the main plant, it will not mature quickly, right? Then gibberellic acid will also speed up malting process in brewing industry, right? What is the meaning of malt? Malt will be produced, it's a raw material, right, for making alcohol, right? It will be used as raw material. So what is this malt? There are certain cereal crops, right? We have to soak the cereal seeds, जैसे कि let's suppose wheat seeds, right, or barley seeds. We will sow them, right? Sorry, we will soak them in water, and we'll see that when they will germinate, a small seedling will come out. We'll dry them, right? Up for seed germination, we need gibberellic acid or not, right? So for seed germination, we need gibberellic acid. Gibberellic acid will produce that small seedling. As soon as that small seedling is produced, the sugar which is present in the cereal from starch, it will change into malt. Sorry, maltose, right? It will change to maltose, and then we will dry it up, right? And then we will use that as a raw material for making alcohol for fermentation, right? So that is called malting. If we are having gibberellic acid synthesis in those seeds, the seed germination process will increase, or it will get promoted, and as a result producing malt will become very easy. So it will speed up malting process, right? Now it increases the length of stem and yield by 20 tons per hectare in sugarcane, right? So I told you na, elongation it is doing, it is increasing the length of grape, it is improving the length shape of apple, it is also responsible for increasing the length of a stem of sugarcane, so that we can increase the produce of sugarcane 20 tons per hectare, right? Then spraying juvenile conifers with gibberellic acid has hastens the maturity period. अब देखो what is conifers? Conifers means we are talking about pinus, right? Citrus, and pinus and citrus, they grow very slow. They belong to gymnosperm. Their growth is very, very extremely slow. So if they are growing very, very slow, right, they will attain their maturity period, or you can say when they will produce cones in which seeds are formed, because gymnosperms are seed bearing plants, right? Pinus seeds are edible also. So if they are producing seed, right, then it will take a lot of time, almost 15 years, right? 15 years. So what we can do, we can spray these particular conifers by gibberellic acid, and it will increase the maturity, like it will uh, hasten the maturity period. Maturity period will come very fast, very quickly, right? Now promotes bolting in beet, cabbages, and many plants with rosette habit. Very, very important, right? What is the meaning of bolting? Bolting means just prior to flowering, there is a rapid elongation between the length of internode, right? जैसे कि you all will see cabbage, right? Cabbage will be like this, not exactly like this. Or you imagine any plant which is growing with rosette habit, all the leaves are condensed together, right? Means all the nodes are present at near to each other, right? Up, have you ever seen flower in cabbage? No, right? Because before that you will harvest it. It's a biennial plant, right? Within 1 year you will harvest it. If you allow it to be on that particular uh, you can say, if you allow it to grow, what will happen? You will see there will be rapid elongation between the internode, right? Internodes are very close to each other. Now here it got elongated, and all the auxiliary buds will get separated, right? And then you will see flowering will occur, right? So this process is called what? This process is called bolting. Okay, rapid elongation in internode prior to flowering. And this does not happen in all the plants. This happen only in cabbage, beetroot, right? Flowering, right? In only in rosette habit type of growth. अब इसके लिए who is responsible here? Gibberellic acid is responsible, right? Now name the plant growth regulator which upon spraying on sugarcane crop increases the length. That is gibberellic acid itself, right? This was asked in 2020. Okay, first statement they are saying that gibberellins cause apples to elongate and improve their shape. Is this correct? Yes. Growth at a cellular level is principally a sequence of increase in amount of protoplasm. Sorry, consequence of increase in amount of protoplasm. Yes, cellular level pe growth is when protoplasm is increasing, right? So here both the statements are correct. Both 1 and 2 are correct, right? Then what would happen if gibberellic acid 3 is applied to rice seedlings? Okay, if gibberellic acid 3 is applied to rice seedling, plant show extra elongation, length of the plant decreases, it leads to apical dominance. We can only consider this, right? Because one of the major role of gibberellic acid is to allow elongation, right? Internode elongation just prior to flowering in rosette plant like cabbage and beet, this occurs because of gibberellic acid, right? Now, the third hormone, the most important hormone, that is cytokinin. Cytokinin name itself is telling you about the next chapter's topic, that is cytokinesis, right? It is cell division promoting hormone. So it is also a kind of plant growth promoter. Skoog and Miller, they crystallized a cytokinesis promoting active substances, and they called it as kinetin, right? Kinetin is a modified form of adenine which is formed from an autoclave herring sperm DNA. What is this herring sperm? Herring is the name of fish, right? This F. Spoog and Miller, what they were doing, they actually isolated the DNA from this fish sperm. Okay, herring ka sperm DNA they have isolated. They have autoclaved that DNA. Autoclave is a device where you will where the temperature is very high. You actually clean the instruments and all which are used in surgeries. So that is a boiling hot temperature. In that, in a test tube, he has boiled the DNA from this fish sperm DNA. Sorry, fish sperm. And then what has happened? The particular DNA has got denatured, broken, and one of compound from that DNA, adenine, it gave rise to another compound that is called kinetin, right? So kinetin is never occurring naturally in animal also. It is formed by autoclaving of this herring sperm DNA in a lab. Okay, it's artificial. It is animal based cytokinin, right? So that's why I have wrote it does not occur naturally in the plants. Now, the similar type of cytokinin is obtained from maize plant, that is called zeatin. It occurs naturally in maize. Also, you will find this in coconut milk, right? So, what they found about this particular cytokinin, whether it is zeatin, whether it is a coconut milk, corn kernels, right? What they have done, they have taken corn kernels, they have crushed that coconut milk, they have taken vascular tissue extract, right? Vascular tissue extract, right? And they have added this all in the cells which are dividing, right? When they added all these things, okay, either a DNA extract or vascular tissue extract or coconut milk or corn kernels, they saw that it was increasing the cell division capacity of the cells which are kept in the petri dish, right? You assume what they have done, they have taken a petri dish, there was some cells which are extracted from the pith cells of tobacco plant. One tobacco plant is there, usme the central region is called pith, that is made up of parenchyma cell. They extracted those cells, kept on the petri plate, and they added nutrient medium and everything, and they added corn kernels or coconut milk or vascular tissue extract or DNA extract, and they saw that the cells started dividing, right? So they understood that these things are containing some cytokinesis promoting activity wala chemical that is called cytokinin, right? So what all roles this cytokinin can perform, what all it things it can do, it can do so many things which are related with in like making new organs and all. But you have to remember it will be synthesized in all such areas where rapid cell division is occurring, right? Like root apices, developing shoot buds, young fruits and all, because here continuous cell division is necessary. It helps to produce new leaves, right? Obviously it is an organ. If you want to make a new organ, you need tissue and cells. Cells are formed by cytokinesis. Who will promote it? Cytokinin, right? So it will be making new leaf. You have to remember within these new leaves, chloroplast also will be formed by the same pigment. Sorry, chloroplast is also formed by the same hormone. Then it will be responsible for the growth and development of lateral shoots, right? Remember apical dominance, because of auxin lateral shoots are not developing, right? Because it was inhibiting the cytokinin. So cytokinin actually plays a very important role in producing lateral shoots. Adventitious shoot, what is the meaning of adventitious shoot? Adventitious shoot means a shoot which is formed other than plumule. Okay, let's suppose I am saying here I have a petri dish, I have kept some of the cells from put that from these are some pith cells, okay, from tobacco plant. Okay, these cells are parenchymatous cells, so they cannot divide. But here if I have cytokinin in high concentration and auxin little less than cytokinin, you know what will happen? This particular cells will start dividing, and you will see there will be shoot formation from these cells, right? अब देखो, this shoot is not getting formed from plumule, right? So you will call this shoot as adventitious shoot. Understood? Now, if I change the concentration, if I decrease cytokinin, And if I increase auxin, after some time you will see there will be root formation. This form shoot, this forms root, right? But both are adventitious in nature. They are not formed from radical or plumule. They are formed from some other part, right? So based on the relative concentration of cytokinin and auxin, I can develop adventitious shoot or root. If you want to develop adventitious shoot, you need more cytokinin synthesis, right? It overcomes apical dominance. Obviously, apical dominance will be overcome by cytokinin, right? Auxin effect can be removed by cytokinin. It promotes nutrient mobilization, very, very important, right? How you can understand this point? Let's suppose this is a mature leaf and this is young leaf. Okay, in this young leaf, definitely cytokinesis will be going on, right? So young leaf mein to naturally there is cytokinin, cytokinesis is occurring, cytokinin is there. So all the nutrients which are absorbed from the roots, they will always go towards cytokinin, right? And if nutrient supply is there, it means growth is occurring. So cytokinin presence will be an instruction to the nutrients to move toward that side only where growth is occurring. If this is a mature leaf, यहाँ पे तो cytokinin is not present, right? Here if it is a young leaf, cytokinin will be present here. So nutrients will be mobilized towards this side. If I want to delay senescence, definitely if this is yellow, it will do abscission, right? If you want to delay its senescence, right, what you do, you spray cytokinin here. Then the nutrients will think cytokinin is present here, they will start moving towards this old leaf also. And what will plant understand? Plant will understand that still nutrients are mobilized towards the old or mature leaf. It means still some growth related process is occurring. Do not allow senescence, right? So I have written here, it helps in delaying the leaf senescence, it promotes nutrient mobilization. Are you able to understand? Right, this is very, very important. Mature leaf may to cytokinin is not there, that's why senescence will take place. But if we spray if we spray cytokinin there, plant will get a instruction that okay, nutrients must be mobilized towards this side, because maybe cytokinesis is occurring, and as a result the senescence will be delayed. Otherwise, if cytokinin is not present naturally here, no nutrient mobilization occurring, it should get senesced very quickly, right? Now, kinetics was first discovered from kinetin, they are writing, not zeatin, right? So you have to remember it is autoclaved herring sperm DNA. Naturally, it does not occur in plant. Cytokinin help in delaying leaf senescence. Yes. Reason, cytokinins promote nutrient mobilization in the older leaf. Yes. So both the reason, both assertion, reason is correct and reason is correct explanation for the given assertion. Cytokinin help overcome. Acha, you have to find the statements which are incorrect, right? These kind of questions are very big trap. They will give everything and you have to find incorrect only. Okay, sometimes we'll think it's just asking us to find correct. So cytokinins help overcome apical dominance. This is correct, true. Kinetin was discovered from coconut milk. This is false. What was the name of that which was discovered from either maize or coconut milk? It was zeatin, right? Zeatin does not occur naturally in plant. Wrong. Kinetin does not occur naturally in plant. Cytokinins help in delaying the leaf senescence. Yes, by doing nutrient mobilization. So what's the answer? 2 and 3 is the answer. These are the two incorrect statements asked. Okay, now we have ethylene, right? Ethylene was discovered by H.H. Cousins. Ethylene was discovered when he saw that when ripened oranges were kept near the unripened banana, some volatile substance was moving from these oranges to banana, and it was allowing the ripening in banana. So this volatile substance was later found to be ethylene gas, right? So this ethylene gas is synthesized in very large amount in those tissues which are undergoing senescence. Okay, and in the fruits which are undergoing ripening. So senescence and fruit ripening के लिए this is very important hormone, right? Then you have to remember there are three things which you have to remember, which will be shown if any particular plant is producing ethylene. That is, during the growth of seedling, there will be swelling of the axis of the seedling, right? There will be formation of apical hook, right? Okay, these two things, and horizontal growth of the seedling, right? These are the three things which you will observe during the seed germination in the growth of seedling, which will be responsible that this is happening because of the influence of ethylene. So horizontal growth of seedling matlab usually seed will grow like this vertically, right? But here the seed will grow horizontally for some time, right? The axis of the seedling will swell, and you will see there will be a apical hook formation like this. Plumule will be present like this. This plumule will be present in apical hook, so that the shoot apical meristem should not touch directly the soil particles, and it should not get damaged, right? Otherwise what will happen, pathogens can enter. So it is protected like this. This happens because of ethylene synthesis in the seedling, right? So these three things together are called triple response, right? If anyone wants to check that whether ethylene synthesis is occurring in the plant or not, they can check with the three different responses of ethylene, that is horizontal growth of seedling, okay, swelling of the axis of seedling, and apical hook formation. Now what's the major role of this hormone? It will promote senescence and abscission in the leaves and flowers. If they are mature, right, they should be dropped off from the plant, right? That is called abscission. And if it is mature, they should if it's fully mature, they should be detached from the plant, they should be dead. So it's called senescence, right? So promoting senescence and abscission in the plant, it's very important for fruit ripening, right? And it increases the ripening in the fruit by increasing the rate of respiration, right? This sudden increase in the rate of respiration is called respiratory climactic, right? And such fruits in which rate of respiration is increasing very quickly due to ethylene synthesis, that is called climactic type of fruits. There are two type of fruit, climactic or climacteric and non-climacteric fruits, right? Climacteric fruits will be of that kind. When you will remove that particular fruit from the parent plant, they will further not ripe, right? Once they are present within the plant, attached to the plant body, they are ripening at that time because of heavy synthesis of ethylene, right? After that they will not further mature. If they will not further mature or ripen, then they are called non-climatic. If they are ripening very quickly under the influence of ethylene, when they are connected to parent plant, they are having high increased rate of respiration, then they are called climactic type of fruits. Only this much you have to remember. You don't have to do anything else which fruits come under climactic and all. That is not required, right? Now what's another role of ethylene? It breaks seed and bud dormancy. Very, very important. Seed dormancy is broken by ethylene, bud dormancy is broken by ethylene, right? So you have to remember this point. Initiates germination in peanut seed. This you have to remember, right? Some examples are there in different hormones. You have to remember them. Peanut seed may initiate germination. It will be responsible for sprouting in potato, right? So sprouting of potato tubers. It promotes rapid internode or petiole elongation in deep water rice plants, right? This is also very important. If you consider this point, elongation in the rice plant is coming because of, like, because of ethylene, and you have learned that mostly elongation is done by gibberellic acid. So let me tell you one concept here, okay? This rice plant, when it is in the seedling stage, okay, this rice plant when it is under the seedling stage, it is in the water, right? And it is producing what? It is producing ethylene. And you have to remember, ethylene is actually inhibitor of abscisic acid, and abscisic acid is an inhibitor of gibberellic acid, right? So if any plant, any rice plant is under the water, this ethylene is a gaseous hormone. It cannot be dissolved in water. It will be limited in the plant body itself. So its concentration in the plant body will increase, and as a result what will happen, it will inhibit all the abscisic acid. If abscisic acid is inhibited, gibberellic acid will be uh, removed from the control of abscisic acid or not? Now gibberellic acid is there, right? So what will happen? Elongation will occur or not, right? So actually who is responsible for elongation here? Also gibberellic acid. But because of what? Because of ethylene. That's why you have to remember ethylene only, otherwise there is no use of gibberellic acid, right? Because it's under the negative impact of abscisic acid. So it is promoting the rapid, it's not written it is responsible, it is written it is promoting the rapid internode and petiole elongation in deep water rice plants, right? Then it promotes root growth and root hair formation, very very important. Root ka growth, root hair development, this all is because of ethylene. Initiates flowering and help in synchronizing the fruit set in pineapple, right? Who was responsible for inducing flowering in pineapple? It was auxin. Who is responsible for initiating flowering and synchronizing the fruit set in pineapple? That is ethylene. It also induces flowering in mango, right? So they already have asked a question in PYQ 2 years before on this. Then there is one aqueous solution of this ethylene hormone that is called ethiphone, right? Because you cannot use a gaseous hormone if you want to have same applications seen on the plant. So you will be using one aqueous solution of ethylene that is called ethiphone. It will hasten the fruit ripening in tomato. It will increase the ripening of tomato, apple, and it will accelerate abscission in the flowers and fruits, right? Because it will definitely accelerate senescence, abscission, all of these things. And when the fruit is ripening, definitely there is a lot of changes in that fruit. Pericarp will change, right? It will become thin, right? The sugars will change. So here they have written there will be thinning of cotton, there will be thinning in the fruit ball of cherry, there will be thinning in the fruit ball of walnut. You just think that if the fruit is raw or not fully ripened, right? Think about cherry or tomato, right? It will be having thick fruit wall. But if the fruit is very ripe, what will be happening? The fruit wall will become very membranous, right? Pulp will be developed. So in cotton, cherry, walnut, thinning of the fruit wall will be done because of ethiphone application. And it also will be responsible for promoting the female flowers in cucumber plant. Cucumber plant is a plant which belongs to family Cucurbitaceae, and it produces unisexual type of flowers, right? So unisexual matlab it is having male flower and female flowers, right? अब you tell me, if you want more fruits, more cucumber, which flowers are needed more in number? It is female flower, right? So ethylene is promoting synthesis of female flower so that we can increase the yield of cucumber. Are you able to understand? Right? So these are some of the things which you have to remember. Now you can solve these questions. Ethylene is used for what? It is used for delaying ripening of tomato? No, not delaying. It is going to increase the ripening, right? It is hastening the ripening of fruits? Yes. It slows down ripening of apple? No. So option B is the answer, right? Here, ethiphone in an aqueous solution is readily absorbed and transported within the plant and releases ethylene slowly. Yes, this is correct, right? Cytokinin promote female flower in cucumber. This is wrong, right? Ethiphone will be absorbed by the plant and it will release it slowly, slowly. That is correct, right? But cytokinin will promote female flowers. No, it is ethylene only. So statement 1 is correct, but statement 2 is incorrect, right? Okay, it takes a very long time for pineapple plants to produce flowers. Which combination of hormones can be applied to artificially induce flowering in pineapple plants throughout the year to increase yield, right? So you know the two set of hormone which are responsible for pineapple, right, flowering, that is auxin and ethylene, right? They have given this question in 2019, right? Now the last hormone for the chapter, right, that is abscisic acid. So abscisic acid is actually plant growth inhibitor, right? So what are the inhibitory effects of abscisic acid? Name itself is telling it is responsible for abscission, right? That leaf drop, fruit drop will be promoted by abscisic acid, and it will also induce dormancy in the seed, right? It will not allow the seed to germinate. Seed will enter in the suspended period of growth. It is in general, okay, okay, it is a general plant growth inhibitor or inhibitor of plant metabolism. It inhibits the seed germination. So with this we can say it is just opposite to gibberellic acid, right? It is antagonistic. It stimulates closure of stomata, right? Whenever there is any water stress, right, whenever any condition where water is very less, at that time what will happen, abscisic acid will be produced, and abscisic acid will be responsible for closing of the stomata. That's why it is also called sometimes stress hormone, right? So it stimulates the closure of stomata. It plays very significant role in the seed development, maturation, and dormancy. I told you that seed dormancy is not bad for the plant. If the seed has not developed properly, if the embryo has not matured yet, if the seed coat is still getting formed, right, then we should not allow the seed to germinate. Otherwise, if underdeveloped type of seed germinates, what will happen? It cannot produce a healthy plant, right? So at for like elongating the time period, or for extending the time period, right, or for extending the time for maturing the seed properly, seed dormancy is needed. So in a way we can say abscisic acid is having negative impact on seed germination, because it is not allowing the seed to germinate. It is adding up in dormant period, right? But this occurs to allow the seed to develop and mature perfectly, right? And if the conditions outside are perfect, well and good for the seed to germinate, then it can germinate. Okay. By inducing dormancy, ABA helps seed to withstand desiccation. Let's suppose conditions are dry outside, moisture is not there, right? Suitable temperature is not there, oxygen is not there. Then in unfavorable condition, definitely seed cannot grow. So in that case also ABA will help. So I told that if like let's suppose seed from within, if there is any problem, then also ABA can help in extending the development period and maturation period by allowing dormancy. Or if outside conditions are unfavorable, then also abscisic acid may help, right? In most of the situations, ABA acts antagonistic to gibberellic acid, right? It's just opposite of gibberellic acid, right? So this is what you have to remember. Question also is on the same line, that ABA is antagonistic to gibberellic acid. Okay, to avoid excessive water loss during severe drought stress, the closure of stomata is signaled by the production of which particular type of hormone? It is abscisic acid, right? So this one, which of the following events in plant is or are affected by interaction of more than one PGR? Ab dekho. This question is based on a topic which is called, uh, you can say how these PGRs are interacting, right? There is some of the PGRs which have synergistic effects. Synergistic effects. Some have antagonistic effect. Okay. Synergistic effect is with respect to a particular function. If two hormones are involved, both have positive impact, right? Antagonistic means if two or three hormones are involved, right? Two hormones we can like clearly understand. So one is having a positive impact, another is having a negative impact, so it's opposite to each other, right? With respect to one function. Okay. Just say how you can understand this. You can understand with respect to flowering in pineapple, both auxin and ethylene are synergistic. Understood? With respect to seed germination, both gibberellic acid and abscisic acid, they are antagonistic to each other, right? So here they are saying which of the following events are affected by interaction of more than one PGR. For apical dominance, only auxin is required. For abscission and senescence, we can have which all hormones? We can have ethylene also, and we can have abscisic acid also. For dormancy in seed or bud, we can have you can say auxin, sorry, for dormancy in seed and bud we can have abscisic acid, right? So we can take this as an answer, right? But I think you have studied induces seed and bud dormancy in case of ethylene also. So if we take ethylene, okay, with very few examples ethylene will do this work, but abscission and senescence, this will be done by ethylene, sorry, as well as abscisic acid, both. right? So we have to do this. Okay, so this was the last part of our previous chapter, plant growth and development. Now we have to revise one more chapter, that is cell cycle and cell division. I'll give you 5 minutes break, right? You can transition from one chapter to another chapter, and then we'll very quickly try to complete this chapter in a very less time. Okay, thank you. Okay, so I hope you all have joined again. Now we have to discuss cell cycle and cell division. This is also one of the most important chapter of botany, right? You will be getting cell plus cell cycle combined 10 to 12 questions. The number can be more also, because there are so many questions which can be made out of this chapter, right? So basically we all have studied this chapter, I think, from class 9th itself, and we have some basic information about this, right, from our junior classes. Again, this chapter also says the very same thing which plant growth development says, that we all started our life as a single cell, that is called zygote. Now you just think that from that single zygote, making a multicellular organisms like human, it requires lot of efforts, right? So how one single cell converts into multiple cell organism like us, right? Definitely one cell has to divide and it has to give rise to these cells, right? So division is required, cell division is required. Zygotes first develops into a small multicellular structure, that is called embryo, and then that embryo develops completely into a entire organism, whether it is a plant, whether it is an animal, or whether it is any kind of multicellular organism. Even if you consider unicellular organisms also, if a unicellular organism has to give rise to another organism, it also relies on the same process, that is called cell division, right? So cell division is very necessary. It increases the number of cell. It is responsible for multicellularity. It is responsible for the growth. It is responsible for reproduction in unicellular organisms, right? So cell division is what we have to study, right? Now, cell division and cell cycle, do you think this occurs in all the living organisms together, right? Whenever a cell divides, it will follow some event, some sequence uh, of events which will occur in order format, right? Just say, for example, maybe you remember you have studied G1, S, G2, M phase, right? So all these are what? These are the stages of cell cycle. A cell will complete all of these stages, then it will divide. Then again the daughter cells, if they have to divide, they will complete all these stages and they will divide, right? So these are called cell cycle stages. Do you think that a cell divides by completing all the cell cycle in all the organisms? So the answer is no, right? Cell division occurs in all the organism. Cell cycle only takes place in eukaryotic organisms, right? Eukaryotic cells only will undergo cell cycle. These all stages which you study, G1, S, G2, M phase, mitosis, meiosis, all of this exist only in eukaryotic cell. It can be a plant cell, it can be an animal cell, it can be a fungal cell, even it can be the unicellular eukaryotic cell, that is called protist, right? But before that, if you go to Monera, right, in prokaryotes, you will never find any kind of cell cycle working there, right? So cell division occurs in prokaryotic cells, but cell cycle does not occur in any kind of prokaryotic cell. And here I have given one statement. Just please tell me it is true or false. I have written bacteria prepares for cell division in interphase stage. Can I write anything like this? No, right? This is a false statement because I am telling you there is no interphase, no G1S, G2, right? There is no M phase, nothing is there in bacteria. Bacteria divides by binary fission, right? It divides by another method. It does not follow cell cycle to divide. Okay, I hope this is clear. One more type of question can be asked like this: cell cycle does not occur in, right? And you can see here what we have agaricus, which is called mushroom, right? We have penicillium, which is a type of ascomycetes member. We have streptococcus, which is the name of bacteria, and we have mimosa pudica, which is the name of a plant. So tell me, where you will not observe any kind of cell cycle? You will not observe any kind of cell cycle in streptococcus. Even if I write the name of any virus here like TMV, then also it will not take place there, right? Because viruses, they are acellular, right? They are acellular. There is no cell at all. So how they can have a cell cycle, right? Okay. Now you understood about that. Basically, we have two types of cell divisions, which we all know. The most common one which is there in most of the organisms, that is called mitosis, right? So cell division can be divided under two category. The most common one, that is mitosis, and the one which is limited to only certain places, and most commonly it occurs during reproduction uh, when the diploid cell has to give rise to gamete, right? When gamete formation has to take place, there only you will see meiosis or reduction division occurring. So you can see here what I have done. I have made a parent cell with 46 chromosomes here and 46 chromosome here. Let's suppose both of these parent cells are starting to divide, but one parent cell, you can see it has divided and it has given rise to two daughter cells, right? And both the daughter cells are having 46 chromosomes only, and along with 46 chromosomes, you can see the daughter cells are exactly replica or copy of each other, right? So parent cell divided and given rise to two daughter cells which are exactly copy of parent cell. Without any change this particular daughter cells have formed. This kind of division is called equational division or mitotic division, right? Where the daughter cells formed are two in number and both are exactly same like parent cell, right? This is called equational division. This is the most commonly occurring division. Another, you can see I have started with the same condition of a parent cell, 46 chromosome. It is dividing and is giving rise to total four daughter cells, and in these four daughter cells you can see every daughter cell is receiving only half number of chromosome, that is 23 chromosome each, right? So 23 chromosomes in this also and this also, right? Daughter cells are four, but it is getting half of the number of chromosome, right? So can I say this is meiosis or reduction division? So at least the basic idea you have got. First we have to study this kind of division which is commonly occurring, then we have to study this kind of division, right, which is only limited up to certain places in our body where gamete formation takes place, right? I hope it is clear. Okay, now what is cell cycle, right? So cell cycle is nothing but its sequence of events. Okay, multiple events are going to occur here in a cell which duplicates its genome. It is responsible for synthesizing other constituents of cell, and eventually it is going to divide the cell into two daughter cells. So sequence of events by which a cell is going to duplicate its genome, right? There are so many sequence. First you will see cell will copy its genetic material, DNA. It will copy other components of it, right, like endoplasmic reticulum, Golgi complex, uh, everything. And then finally you will see it will give rise to two daughter cells, right? So this is the definition of cell cycle given in NCERT. What is the meaning of genome? What is the meaning of duplicate its genome? Genome means all the total haploid content of the cell, all the total genes, DNA, whatever is present in a haploid cell, that is called genome, or total genetic makeup of a cell. It has to copy entire DNA, right? Okay. What are the features of cell cycle? I hope this is visible to you, right? So there are certain features. All the steps which we are going to study, they all will be present in a sequence. right? Just say, for example, what are the sequences which we have to follow? We have to follow that first growth is occurring, right? Then DNA replication is occurring, then only a cell is going to divide. So if I talk about this growth step, right, what will happen in this growth? There will be increase in the cytoplasmic constituent of the cell. The cytoplasm of the cell will increase, the size of the cell will increase. This is the meaning of growth. This is a continuous process, it will occur continuously throughout the cell cycle, right? But DNA replication is going to take place only in one fixed phase, that is called S phase, right? So you have to remember, growth followed by DNA replication. Growth is occurring continuously, DNA replication is occurring in a fixed stage, is a fixed process. Finally, after completing this, the cell is going to divide in M phase, right? So this is a sequential step. Now, the events of this cell cycle are coordinated, right? For example, these fixed changes which are occurring, these fixed steps which are occurring, they are of fixed duration, right? And only after completing one change, next change will be there. Don't think that this is a, like, this is different you can say phases are there. No, a cell is acquiring some changes which are visible to you, right? After completing those changes, next change can be acquired. This is what we have to understand, right? And each and every change is coordinated with each other, right? First we will duplicate the DNA, then we will change the DNA into chromatin, right? First we will not change the DNA into chromatin, it's like this. First we have to divide the nucleus, then only we can divide the cell. So each and every step is actually highly coordinated, right? And all this coordination is brought about by certain genes, right? So all these coordinated steps are actually controlled by genes. It is genetically controlled mechanism, right? Now, there are two phases of cell cycle. If I consider cell cycle, we have two phases: interphase and M phase. Okay, interphase is the largest phase of this cell cycle. Okay, for most of the duration you will see the cell will be in the interphase. This is also known as resting phase. Okay, resting phase means cell is not resting, actually cell is doing lot of work here. Cell is resting with respect to division. There will be no division in this phase, cell will not divide, cell will only prepare itself for division, right? So with respect to cell division, it is called resting phase, right? It is also called preparatory phase. Preparatory phase means biosynthetic phase. Ab dekho, if you want to cook something, you need to gather so many raw materials together, right? So many things are needed to be present together, then only you can make something. So if you want to divide a cell, first you need to make necessary arrangement for that, right? You have to copy so many things, you have to divide so many, like you have to arrange so many things. That is going to happen here. You have to get energy, right? A lot of ATP is required, right? There are a lot of biosynthetic like nucleotide synthesis should be there, because in this you have to make DNA. So उसके लिए you need nucleotides, that all have to be synthesized here. So the cell is actively preparing itself so that it can divide later, right? So in interphase you have to remember, preparation is occurring, preparation to divide is taking place. That's why it is called biosynthetic phase, right? And in M phase, what is going to take place? M phase mostly, because common type of division is mitosis, that's why M phase is mostly called mitotic phase. Here actual division will take place, here one cell will become two, right? It's the most dramatic stage of cell cycle. Why it is called most dramatic stage of cell cycle? Because reason I have given as our cellular reorganization takes place. Maybe you remember, you have studied M phase as prophase, metaphase, anaphase, telophase, right? So in prophase the cell will look something different, in metaphase it will look different, anaphase it will look different, telophase it will look different, right? Lot of cellular reorganization is going to occur, lot of drama is going to take place in the cytoplasm. That's why it is considered as most dramatic phase of cell cycle. Now, what all things are biosynthesized here? So you have to remember, DNA synthesis is going to take place, RNA synthesis is going to take place, protein synthesis also is going to take place here, right? Cell is transcriptionally, translationally active, right? Transcription means RNA synthesis, translation means protein synthesis. Both of these components are getting synthesized. Cell is biochemically very, very active, right? It's not at all resting. It's not dividing. That's why it is called resting, right? cell organelles are getting duplicated. All the cell organelles are getting duplicated here, right? ATP requirement is very high. So there is lot of ATP synthesis. And what is the duration of this stage? Interphase. Interphase will be there for almost 95% of the total duration of cell cycle. Just think by yourself, if you want to make something, the preparation and arrangement of that particular thing will take up a lot of time, right? But actual cooking time or making time or maybe drawing time is very 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 less, right? So whatever activity you are planning takes a lot of efforts, right? So same thing, you are actually preparing here. And the day, like for example, you can understand very easily, there are so many days for which you have prepared for your NEET exam, right? Which is coming after within 2 weeks, right? So just 2 weeks are left, and after that you will be writing the day for which you are preparing. So that day is only one day which is of only 3 hour. So that 3 hour is equal to this M phase, very small, only 5% of the total duration of the cell cycle, right? But for this, that 3 hours, 5%, you have prepared for 95% of the total duration, right? That is the thing which you can remember. Up here, this M phase is further divided into two stages. Karyokinesis means division of nucleus. First, nucleus will be divided, followed by division of cytoplasm or cytokinesis, right? Another question they will always ask, what is the nature of genetic material in interphase and what is the nature of genetic material in M phase? So genetic material in interphase is always of chromatin type, right? Remember, the DNA and histone protein bind together, that will form chromatin, right? So chromatin is a genetic material present in the interphase. And if you come to M phase, right, in M phase you have to remember, in the metaphase, from the metaphase onwards to telophase, you will be seeing chromosome. But in the early prophase and in the late telophase, you will be seeing chromatin, right? So this you have to remember. Now let's discuss about interphase. Interphase is common to mitosis as well as meiosis, right? Whether it is mitosis, whether it is meiosis, preparation is must, right? Whether you give NEET exam, whether you go for board exam, you need preparation, right? So it is must. It's long and is subdivided into three stages: G1, S, and G2, right? Up here I have drawn a simple type of cell cycle stages. So you can see these are the two daughter cells. They will enter into G1 phase, they will go to S phase, right? From here they will go here, from S they will go to G2 and G2 to M, right? Now if you see here, this G2 is called gap 2, G1 is called gap 1. S is what? It is DNA synthesis phase, and M is what? Cell division will take place here. So can you see here that the two main things, right? That is this DNA synthesis and this cell division is having two gap stages, which are gap 1, gap 2, right? So we have to start from here. The divided daughter cells, if they want to further divide, they will enter into gap 1. They will enter into DNA synthesis phase, they will enter into gap 2, and they will further divide into M phase, right? Okay, so let's see what are things which we have to remember in G1 stage. These are the different names of G1 stage. You can call it as post mitotic phase because it is coming after M phase. You can call it as pre-synthetic phase because it is present before S phase. You can call it as gap 1 phase because it's the first gap between cell division and DNA replication, right? This is metabolically active phase. Cell is metabolically very active. Its size is growing continuously, okay, because the cell has to be ready for division, so its cytoplasm should increase. Size is continuously growing. Synthesis of nucleotide is going to take place here because in the replication you need DNA replication, may you need to make DNA. What is the monomer of that deoxyribonucleic acid? It is nucleotide. So here there will be synthesis of nucleotide, right? There will be synthesis of amino acids because you need to prepare a lot of proteins, right? There will be synthesis of protein itself, there will be synthesis of RNA, there will be synthesis of ATP. So many things are getting synthesized. That's why biochemically active phase. Then most of the cell organelles, they are going to be synthesized in this stage, right? Most of the cell organelles, okay, except these Golgi, plastid, and mitochondria. Golgi, plastid, and mitochondria will be synthesized in G2, right? Except these three, all will be synthesized in most of them will be synthesized in G1 stage. Okay, what is S? S is also called DNA replication phase, right? It is DNA replication. It will take place inside the nucleus, right? And when the DNA is getting copied inside the nucleus, the centriole or centrosome in animal cell will get duplicated in the cytoplasm, right? Very important. Then the amount of DNA changes in this phase from 2C to 4C, right? So there is doubling of DNA which is going to take place here. Whether it is 1C, it will become 2C. If it is 2C, it will become 4C. If it is 4C, it will become 8C. So amount of DNA change will take place, or amount of DNA will get doubled here. It will be responsible for doubling of DNA, right? But there is no change in the number of chromosome. Chromosome ka number will not change here, right? Number of DNA, how you are going to count? By counting the number of chromatid. Okay, number of chromosome, how you are going to count? By counting number of chromosome. Okay, in this figure it is very easily visible. Just see this. This is a cell where I have shown you two chromosomes which are same chromosome, homologous you can consider. This is chromosome number 1 ka 2 copy, right? Actually, in the interface, I told you, you will not be able to see any chromosome. What is visible to you in interface? It is chromatin, right? But for simplification, we have to draw it. Otherwise, you will not be able to understand it. Okay. So, you have to assume that actually it will appear like this. But for simplification, I have drawn a diagram where I have shown you two chromosomes, right? Actually, chromatin is visible. Now, let's suppose these are the two chromosomes here. So, I have written what's the number of chromosome? N equals to 2. What is the amount of DNA? For calculating the amount of DNA, you have to count the number of chromatid. This entire structure which you can see here, which we have drawn, this one thing is called chromatid, right? And this thing, this pink thing which we have drawn here, this you know very well, right? This is called centromere. So if you want to count the number of DNA, you will count the chromatin. If you want to count the number of chromosome, you are going to count the centromere, right? So here you can see how many uh, number of chromosomes are there. 1, 2, 2 are there. How many DNA are there? This entire structure 1 and 2, 2 DNA are there. So I can write 2C DNA is there in G1 stage, right? This is G1 stage. Now this DNA synthesis has to take place. In S phase, DNA synthesis will take place, right? And then the cell will come to G2 stage. Okay. In NS phase, DNA synthesis will occur. Then the result we can see is in the late S or G2 stage. So in G2 or late S stage, okay, I can write here this is G2 stage. After the DNA synthesis, I will be having the condition like this, which will actually appear like this, because chromatin, you have to remember, chromosome will not be visible, right? Just count this pink structures, the centromeres, how many are there? Only 2, right? So I have written 2 here. But this structure, how many? One chromatid is there, this second chromatid, third chromatid, fourth chromatid. So total how many chromatid are there? 4. Iska matlab how many DNA are there? 4. So here C equals to 4, 4C. Can you see from 2C it has changed to 4C? Doubling of DNA has taken place. But can you see the number of chromosome has remained unchanged? It is not changed at all. So this you have to remember, right? The DNA doubles, but the number of chromosome after S phase remains same, right? And this is the phase. S phase is the phase where the DNA synthesis also takes place, and there will be synthesis of histone proteins as well, right? Histone protein synthesis will also take place in this stage, because after copying DNA we have to convert it into chromatin, right? Now you tell me the answer of this question. The amount of DNA will transition, or you can say it will change from 2C to 4C during the transition of cell from which stage to which stage? Amount of DNA will change from 2C to 4C when the cell will move from S to G2, right? Because S may, when the cell entered, it was only 2C, right? During the late S phase, it got converted to 4C, right? So the transition from 2C will 4 to 4C will be visible to us when the cell will move from S to G2. Okay. Now G2 stage. because it is coming after S stage, right, and before M stage, right, and as a second gap between DNA replication and cell division, so we will call it as post synthetic phase after S, pre mitotic phase before M, and gap 2 which is present between the DNA replication and M phase. Here also cell will continue to grow. Here there will be synthesis of very important protein that is called tubulin protein, right? Because tubulin protein will help in spindle fiber formation in the upcoming stages of M phase. Prophase may there will be spindle fiber formation, right? So that spindle fiber is made up of microtubule. Microtubule is made up of tubulin protein. So tubulin protein must be prepared now because we need it later, right? Duplication of Golgi, plastid, and mitochondria, right? These three cell organelles which were left there, they will be getting divided in G2 stage, right? Ab you tell me the answer of this question. Cell organelles are duplicated in G1 and S only, G2 and S only, G1 G2 S all, or G1 and G2 only? So this is the answer, right? Most of the cell organelles will get duplicated in G1. Golgi complex, right, plastids, and you can say uh, you can say one more, right, GC, mitochondria and plastid, that will get duplicated in G2 phase. And which cell organelle was duplicating in S phase? That is centriole, right? That will be duplicated in S phase. What about this question? All double membrane bound cell organelles are duplicated in interphase. That is false, right? Can you name me which is that particular double membrane bound cell organelle which is not duplicated in the interphase? Can you please write it down? It is nucleus, right? Nucleus duplication will take place in M phase, not here, right? Other than that, plastid will get duplicated here, mitochondria will get duplicated here. Most of the cell organelles are duplicated in G2 stage. This is also false, right? Most of the cell organelles are duplicated in G1 stage, not G2. Okay. Then there is one more stage which is not a part of this you can say sequence, but there are certain cells which do not divide all the time, right? They don't want to divide all the time in animals, just say for example skin cells, for example nerve cells, right? They don't divide all the time. So because skin cell is an example where you have to remember that it do not divide immediately, it will only divide when it is required to divide, right? Because skin cells will be shed and it will be replaced by new cells, right? So once they will complete their lifespan, they will be replaced back. Or red blood cells, when they will complete their lifespan, they will be replaced back. But nerve cells will never be replaced back, so they are not dividing at all, right? So what will happen after completing M phase? The cells will go in G1 phase. From the G1 phase, the cells will exit. They will go to G0 stage. From G0, they will enter into uh, uh, like in that G0, they will be permanently residing, right? Those cells which never have to divide, they will be present in G0 permanently. They will be arrested in G0. They will never come out of G0, right? But the cells which can divide when required, not now but maybe later, right? They can stay in G0 for temporary time, right? And when they are called for cell division, they will again enter in the cell cycle in the G1 stage. Understood? So it depends on the cell type whether a cell has to stay in permanently arrested condition or the cell has to divide, right, in future. Okay, so I have mentioned here the cells in this stage, they are not dead. You have to remember, they are not dead at all, right? They are not dead. They are metabolically active cell, but they cannot proliferate. They will not divide, right? They will be sitting in G naught only. They will not divide, they will not dead, but they are metabolically active cell. Their metabolism is so slow. That's the only thing. Once instructed, some of these cells can come back and they can enter in G1 and they can resume the cell cycle. Understood? This is called quiescent stage or G naught stage, right? Tell me the answer. Among eukaryotes, replication of DNA takes place in S phase, right? Very easy question. Then match the following list one with list two. They will always give a question like this. Very easy question. Please do it. Okay. M phase is what? It is equational division, because I told you commonly M phase is called mitosis. And what is another name of mitosis? Equational division. G2 phase, what will happen here? Can we say it is an interval between mitosis and initiation? No, no, that is for G1, right? It is interval between mitosis and initiation of DNA replication, G1. What is G2? G2 is the stage where protein synthesis will take place. Yes, quiescent stage, we can say that it is inactive phase. Inactive phase with respect to division. Okay, cell is not dividing, but we cannot say cell is inactive or its cell is dead. It is a phase where cell is not dividing, right, at all. Okay, so what is the answer? We have got the answer option number 3, I think, right? What about this? Which of the following is a quiescent stage of cell cycle? It is G naught, right? What about this? A fruit fly has 8 chromosomes in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, in G1 phase we have 8 chromosome, right? What would be the number of chromosome after S phase? So chromosome number will not change, right? In G1, if it is S, sorry, if it is 8, then in S also it will remain 8 only. Okay, then the centriole undergoes duplication during, right? So centriole duplication will take place in S phase, right? In the cytoplasm or nucleus, please write it. This question, can you do it by yourself? It's just same, right? It's okay, it's same question. Okay, it's a repeated question. Okay, my mistake, I think I have copied this question. Okay, identify the correct statement with regard to G1 or the gap phase, which is correct one. Reorganization of all cell component takes place. No, this is for M phase. Cell is metabolically active, grows, but does not replicate its DNA. Okay, that is correct. Nuclear division takes place. No. DNA synthesis or replication takes place. No. So the most correct or appropriate given option for G1 is only option B. Some dividing cells exit the cell cycle. They will enter vegetative inactive stage. This is called quiescent. This process occurs at the end of, right? So it will occur at the end of G1. From G1 they will enter in G0, right? But yeah, first they will be in M phase, they will enter in G1. From G1 they will enter in G0. Okay, but the thing is that this is not end yet, right? This is called quiescent stage. This process occurs at the end of. Okay, there is a difference in the statement, right? If they are writing end of, then we should mark M phase, because M phase has completed, right? M phase completed. Okay, then the daughter cells are shifted to G1. Up G1 is not completed, right? From here they will be going in G0. If they have to reside there permanently, they will stay there. If they have to come back, they have to come back to G1 and complete G1 and go to S, right? So in that case, because it is mentioned this process occurs at the end of, then we have to choose M phase, right? So we have to check the question, right? Okay, now interphase we have completed. Now we have to learn mitosis or M phase, which is very very easy. Okay, now entire mitosis we can divide under karyokinesis and cytokinesis. Karyokinesis is further subdivided into prophase, metaphase, anaphase and telophase, the four different substages. And cytokinesis, the division of cytoplasm or division of the cell at last. If it is a plant cell, it will be occurring by cell plate formation. If it is animal cell, it will occur by cell furrow formation. Okay, now see, if you consider M phase as mitosis, there are certain points which you have to remember, that the parent cell will divide. It will give rise to two identical daughter cells. Both the cells will be exactly copy of each other as well as a copy of parent. So it is equational cell. Where all places you can see it is going to take place, it will occur in the somatic cells or body cells of animals. It will occur when the embryo is getting formed from zygote, and it will take place in the meristematic cells of plant, right? Now we have certain uh, points to remember for karyokinesis ka first stage, that is prophase. If you see these diagrams, you can understand each and everything very clearly. This is the thing which you have to remember. This is the kind of cell which will start from G2 stage. This cell will enter in prophase. So in prophase, initially the cell will appear like this, right? After completing G2 stage, the cell is going to enter into the first phase of the karyokinesis, that is prophase, right? You can see here by that time the cell entered in prophase, the centrioles which have been duplicated in S phase are present here, right? You can see the intertwined DNA is present here, right? The DNA is you can say coiled together. Coil means it is you can say just intertwined together, right? uh, okay, chromatin material is there. It appears like a ball of wool or not, because lot of DNA will be present to get tangled, right? So this tangled condition of DNA is called ball of wool or spireme stage. In prophase, you have to remember this is the initial prophase, right? Or we can write it. But this kind of diagram is not given in NCRT. Okay, this will be the condition. Just the cell which will enter, it will look like this, right? The cell organelles have been duplicated, everything will like this here, right? Up this cell, once prophase will begin, what will be the condition? This is the NCRT diagram which is given, right? In NCRT, what they have given, as soon as the cell from G2 is going to enter into the prophase, you will see that the chromatin condensation will begin, right? So chromatin, after getting condensed, it will appear like this, right? Can you see? You can see the chromosome type structures there. You can see here, this is one chromatid, this is another chromatid. I'm not saying it is fully condensed, right? But at least something like chromosome is visible to you, which is not visible in this stage, right? So condensation will begin, and ends of the chromosome will become distinct to you, right? Condensation will continue, and the centromeres, sorry, and the centrioles, they will start moving to two opposite poles. This is given as early prophase in NCRT. Now in the late prophase, you will see a condition like this. All those chromosomes which you can see here, they have started condensing. Here you can see they have condensed further, right? So by the end, the chromosomes are looking more condensed. The chromatids and centromere are actually distinctly visible. Here you have centromeres, right? And here you can see chromatids are much more distinct and clear as compared to the previous one. Condensation will continue. Two centromeres, right? They come to opposite poles by the end. The centromeres which have started moving, now they have come to two opposite poles. There will be formation of asteral rays, right? So you will see asteral rays will be formed from here. Now these two centrioles together with as like this centrosome, entire thing, right? Which include these two centrioles and the cytoplasm. So centrosome plus these astral rays together is called aster, right? So aster formation is there. Now these two asters along with spindle fiber, okay, it is called mitotic apparatus. So what is mitotic apparatus? You will see now there will be dissolution of nuclear membrane, right? Nuclear envelope will be dissolved. It will be dissolved. Then nucleolus will get dissolved, right? Dissolution of nuclear envelope. Uh, Disappearance of nucleolus, and other big cell organelles, endoplasmic reticulum, Golgi complex, which take up large part of cytoplasm, they will all disappeared. And then you will see there will be formation of spindle fiber. So spindle fiber and aster together is called mitotic apparatus, which is ready to pull the chromosomes to two opposite poles, right? So mitotic apparatus formation will also be there in this. So this is the case. Late prophase may you will be having this condition. Nuclear membrane disappeared, nucleolus disappeared, spindle fibers, they are formed, mitotic apparatus is formed, right? So we can write spindle fibers plus asters, right? Spindle fiber plus aster is called mitotic apparatus, which will be helping in you can say achieving separation of chromosome, right? All of these things will disappear. Chromosome will further condensed, but at least you can see that two chromatids are sticked to one centromere very clearly. Now the cell will move in next stage, that is called metaphase. Before that, can you see one question here? In prophase, chromosome are fully condensed, therefore one chromatid and one centromere is distinctly visible. Can we say that chromosome are fully condensed in prophase itself? And can we say only one chromatid is visible? No, this stage is coming after S phase. So each chromosome is containing how many chromatid? Two chromatid, right? This is false, and it is not distinctly visible here. It will be very fully condensed in the next stage. The chromosome appears to be monad type, in prophase. This is false, right? If I draw a chromosome like this, then this is called monad type of chromosome. But if I draw a chromosome like this, then it is called dyad type of chromosome, right? There is a big difference. Monad type of chromosome is always present in G1 stage, and after S stage only we will be having this kind of chromosome, because whatever DNA was present here, we have made a copy of this DNA. This was the parental DNA, we have copied this DNA in S phase, right? That's why this statement is also false. Now this is transition to metaphase. As the mitotic apparatus is formed, the spindle fibers will try to attach to the chromosome. To which part of the chromosome the spindle fibers they will attach? You know very well, if this is a chromosome like this, okay, this is a dyad type of chromosome. To this chromosome, we have here this structure. This structure is called kinetochore, right? This structure is called kinetochore. Spindles will attach to the kinetochore of the chromosome from both the sides, right? So this near the centromere, this kinetochore is located, which is a proteinaceous structure, right? This protein disc-like structure. The spindle fiber will get attached to both the sides, right? And the chromosome will start moving, right, towards from the pole. It will start moving to the center of the cell, or you can say equator of the cell. In the image you can see it very clearly, all are moving. This movement of chromosome is also called congression. Okay, chromosomes are moving to align at the center. This movement of chromosome is called congression. Now you just see I have mentioned all of that, okay, which is given in NCERT. Disintegration of nuclear envelope marks the beginning of metaphase. As soon as nuclear envelope is disappeared, all the contents are in the cytoplasm, we will say metaphase has started. When the nuclear envelope disintegrates, chromosomes come in cytoplasm. Condensation completes in the metaphase, right? So if they ask in which stage condensation completes, you have to remember it is metaphase. Here the chromosomes are appearing like rod, right? They are short and they are thickest, right? So this is the best stage to study the morphology of chromosome. If you want to study the morphology of chromosome, metaphase is the best stage, right? You can study morphology में number of chromosome, you can study size of chromosome, number of chromatid, position of chromosome, everything you can study very well in the metaphase, right? Now spindle fibers which are attached to a disc like proteinaceous structure, kinetochore. Okay, spindle fibers I told you they're connected to a disc like structure called kinetochore. This is proteinaceous, it is attached on both sides of the centromere. Now chromosome will start moving towards the equator to align in the center, or align in the equator to form a straight single line of chromosome in the equator, that is called metaphasic plate. And this movement of chromosome to come in the center is called congression, right? So now your cell will appear like this. Can you see here four chromosomes? They all have been attached by their kinetochores to the spindle fibers, and they all are coming in the equator or center. This is making a metaphase plate. You can see two poles, right? This is the condition. Up further there will not be any kind of condensation. This is maximum condensation, so you can study the morphology of chromosome in this stage. From each side you can see two spindles are connected, right? Now next phase will be there, that is called anaphase. Now from both sides, if spindle fibers are attached to the kinetochore, now the spindles will pull the centromere, right? If the centromere is pulled, what will happen? Centromere will break. If the centromere will break, then the sister chromatids will be pulled apart to two opposite poles. So splitting of centromere will occur, sister chromatids will start moving to two opposite poles, they will reach to the two opposite pole, and you it will appear like this, that okay, let's suppose here we have kinetochore also. Here also we have kinetochore, and this is our spindle fiber. So what is appearing here? It is appearing that the centromere is uh, at the leading edge, right? The centromere is at leading edge, it's in front, and the chromatids are trailing behind it. It's like trailing behind the centromere, right? Because it is getting pulled to two opposite poles, right? This is how it will appear. We were having total four chromosomes, right? Can you see all the centromeres have split, right? And now this you can see it will be there in, this is our kinetogote, this is our centromere, this is a single chromatid, right? You have to remember number of chromosome in cell undergoing anaphase. Can you count? I told you number of chromosome will be counted by number of centromere. So total how many centromeres are there here? 8, right? In a total cell in anaphase, in mitosis, you have total 8 chromosome. Okay, so doubling of chromosome appeared here for a very short duration, right? But at each pole only 4 chromosomes are there, right? 1 2 3 4, 1 2 3 4, 4 are there at each pole, right? Up just imagine if this is packaged inside 2 daughter cells, definitely daughter cell will receive 4 4, and as a parent cell also we started 4 only, right? This is the particular stage where you can study the shape of chromosome very well when they are trailing. This is V shaped, this is L shaped, this is J shaped, and this is I shaped, right? Now telophase, this is the last phase of karyokinesis, the most important. The chromosomes, they have reached to two opposite poles. Now what will happen? Now they will start getting decondensed, right? They will start decondensation, right? And they will lose their identity as chromosome, and they will change into discrete elements, which is called chromatin, right? That's why I told you in prophase initial part you will not see chromosome, you will see only chromatin. From metaphase onwards you will be seeing chromosome, and after reaching telophase again you will be seeing chromatin, because decondensation will take place here. So chromosomes which have reached to two opposite pole, they will undergo decondensation and they will change into chromatin, right? Now here mitotic spindle work is over. Mitotic spindle was formed so that they can separate the sister chromatid. Now mitotic spindle will disappear. Now nuclear envelope will reappear, nucleus will reappear, Golgi, ER, everything will be reappeared. And then you will see there will be formation of two daughter nuclei as soon as the nuclear membrane is reappeared. So here everything you can see which is occurring is just opposite of prophase. That's why this is also considered as reverse prophase. Okay, sometimes you have to remember this telophase is also called reverse prophase. Okay, and the cell will appear like this. So chromatin have been separated, packaged inside daughter nuclei, right, with one centromere there. Now what should happen? Now there must be cytokinesis, karyokinesis is completed. Now cytokinesis of two type, right? If it is a plant cell which is undergoing cell division, then it is cell plate formation. If it is animal cell which is undergoing cell division, then it will occur by cell furrow formation, right? Now here you can see I have made the diagram here, you can understand very well, right? So here what will happen, you have to remember this will be the condition of the daughter cell, daughter nuclei which will be created, right? Golgi complex will start producing certain Golgi vacuoles which will come and they will align here in the center of the cell, right? From both the sides these Golgi vacuoles will come and they will align in the center, right, like this. And using this as a guide, there will be deposition of calcium pectate here, then there will be deposition of cell wall material here, right? And then there will be formation of complete cell wall. So when these Golgi vesicles are deposited, this forms a structure, and after getting deposited, the plasma membrane of all these Golgi vesicles fuse, right? Then there will be deposition of cell wall material, then it is called cell plate or phregomoplast, right? Cell plate or phregomoplast, right? This divides the cell or not. Here from the center to periphery, if the cell is dividing, right, from center to periphery, this kind of division is called centrifugal division, right? The Golgi vesicles will come and deposit in the center from both the side. It will mark the position, yes, divide the cytoplasm from here, right? All the vesicles will fuse together, right? Plasma membrane will be formed, right? Then what will happen? In between there will be deposition of cell wall material, middle lamella, right? And it will further separate the two daughter cells. Here you have to remember in animal cell what is the condition. In animal cell you see this is called actin filament. Okay, actin filament is a type of protein filament which is always associated with the plasma membrane. So when these filaments will uh, you can say move towards this side, towards the center of the cell, along with that the plasma membrane also will you can say like it will form a furrow or a cleavage, right? And then this cleavage will deepen and it will fuse in the center and it will divide the to daughter cells. So here what is happening, from periphery to center division is occurring. That's why this is called centripetal division. Okay. How all these things will take place, I have given here. You can read it later and you can easily understand what is happening here step by step. Here it is mentioned Golgi vesicle deposit in center, they fuse together, they make the they mark the formation of cell wall. Precursor of cell wall is called cell plate or middle lamella, and here formation of cleavage or a furrow on periphery, it depends and fuses at the center, and it divides and the cytoplasm into two. Okay, this is the significance of mitosis. I hope you know this. Mitosis is the division which is very essential and important for growth of a cell. It is very important for making a complete organism from zygote, right? Cell growth result in disturbing the ratio between the nucleus and cytoplasm. Now imagine the cell cytoplasm is growing, nucleus will be appearing very small or not, right? Therefore it is very important that the cell also should divide, nucleus also should divide, and there must be a nucleocytoplasmic ratio which can be maintained. देखो, in this slide you can imagine, like you can consider it. Here there is small amount of nucleus, okay, sorry, small amount of cytoplasm and nucleus. So here n by c ratio is very high. Nucleus can, this much nucleus can efficiently regulate every activity which is occurring in this cytoplasm, right? But if the size of the cell is increasing and nucleus is same, right? If the amount of cytoplasm is increasing, right, this value is increasing, right, and nucleus is of same size, what will happen? N by c ratio will decrease or not? If n by c ratio will decrease, nucleus cannot fulfill the functions of this entire cytoplasm, right? Then for you can say efficiently controlling all the functions, what should happen? The nucleus should divide and cell should divide, right? So to restore the n by c ratio, the cell divides, right? If the n by c ratio is low, right, then it will result in division or mitosis. Okay. Now, it is very important for cell repair, tissue repair. So, lost cells in the case of injury or wound can be replaced by mitosis. Same cells, if you want to bring back to the body, mitosis is the only way. RBC will be again and again getting formed by mitosis only, because RBC has a lifespan of 120 days. After that, it will die. It will be replaced back by mitosis. Now, reproduction. For simple lower organisms like unicellular organisms like algae and protista, fungi, bryophyte, honeybee like drone, right, which is the male honeybee, they all divide by mitosis, right? Okay, they will make gamete by mitosis. Then you have to remember some of the things, like there is one particular chemical which is going to arrest the mitosis, or you can say it will inhibit spindle fiber formation. If spindle fiber formation is not there, metaphase के आगे anaphase cannot occur, metaphase cannot take place, right? So cell will be arrested in the metaphase. So this can be done if you apply one chemical that is called colchicine, which is extracted from Colchicum autumnale, one of the plant which belongs to family lily, right? Liliaceae family, right? So colchicine is considered as mitotic poison because it is going to arrest the cell in mitosis, right, in the metaphase stage of mitosis. Then there are certain group of chemicals which will you can say promote mitosis, that are called mutagens. Like in animal cell, the most common mutagens are insulin, and in plant cells it is two hormones which you have learned just now, auxin and cytokinin, right? Then unicellular eukaryotic organisms, they divide by amitosis, right? They will not have any stages like mphase and all. Their nucleus will elongate, their cell will elongate, cleavage formation will be there in nucleus, cleavage formation will be there in the cytoplasm, and they will divide. That is called amitosis. In unicellular type of prokaryotes, binary fission is there, that is there in bacteria. Okay, so mitosis only occur in diploid cell. Is this correct? No, this is false. Mitosis can take place in both haploid cell as well as diploid cell. Okay. Now, spindle fibers attach to kinetochore of chromosome during which stage? Spindle fiber, they attach to the kinetochore during the, as per NCRT, it is metaphase, right? Doubling of number of chromosomes can be achieved by disrupting mitotic cell division soon after, right? So doubling of the number of chromosome can be achieved by disrupting mitotic cell division soon after what? Okay, this is from deleted. Yes. If you disrupt anaphase, then what will happen? There will be doubling of number of chromosome or not, right? Because that is the stage. Okay. With respect to metaphase, which of the following statements are incorrect? Right, chromosome lie at the equator of the cell, correct. Complete disintegration of nuclear envelope takes place, correct. Chromosomes are highly condensed. Metaphase chromosomes are made up of four sister chromatids held together by centromere. This is wrong. You have two sister chromatids held together by one centromere. Select the incorrect statement with reference to mitosis. Okay, all the chromosomes lie at the equator at metaphase. Splitting of centromere occur at anaphase. Spindle fiber attached to centromere of chromosome. Chromosome decondense at telophase. Right? So, what is incorrect with respect to mitosis? Splitting of centromere occur at anaphase. This is correct. All the chromosomes lie at the equator at metaphase. Correct. Spindle fibers attached to centromere of, okay, so this can be somewhat taken because spindle fiber will attach to the kinetochore. Okay, chromosome decondense, that is also correct. Okay, attachment of spindle fibers to kinetochores of chromosome becomes evident in which stage? Attachment of spindle fiber to kinetochore of chromosome becomes evident in the metaphase. Okay, which of the following is not a characteristic feature during mitosis in somatic cells? Synapses will not occur in the mitosis, right? This is a concept of meiosis. Spindle fiber attach onto kinetochore of the chromosome, right? So you know these questions are very easy questions. That's why it's taking 1 or 2 seconds to do, right? You can pause the video and you can do it by yourself if you want. Time limitation is there, so I am doing it very fastly. Okay, now the last one, that is meiosis, the most important one and the most technical one. Many students will not understand it, so we'll take up some time in this. We'll try to complete it, right? So meiosis is what it is, actually reduction division. Just now we have seen in first slide, 46 chromosome will give rise to 4 daughter cells, right, which is having 23 chromosome each. So it's a reduction division. It results in decreasing or reducing the number of chromosome in the daughter cell to half. Where all places it will take place? It will most commonly take place in the reproductive cell or germ cell of a diploid organism. If any organism is diploid like humans, right, and if it is having total chromosomes as 46, right, so in the gametes, male gamete there will be 23 chromosomes. In the female gamete also there will be 23 chromosomes, right? So here n and n, right, this is going to take place in humans. This is for gamete formation. That's why it is called gametic meiosis. This is the most common and this is the only one which we have to study. But do not forget that there are certain plants like bryophytes, like pteridophytes, like gymnosperms, even in angiosperm. Meiosis will also take place in order to form a spore. That is called sporic meiosis, right? And then there are certain green plants, like certain lower group of plants like green algae and certain protists, where you will see meiosis will take place in a zygote to form haploid spore, right? So there are three places where meiosis can take place which are given in NCERT, but we have to study gametic meiosis in this chapter. Okay, now here you can see I have given one brief account here. Okay, number of daughter cells which are produced after meiosis are 4, and the daughter cells will not be identical to the parent cell, and the daughter cells will not be identical to themselves as well. This is the parent cell which is diploid. You can see meiosis is divided into two type, like two steps, meiosis 1 and meiosis 2. So after meiosis 1 itself, you can see the two daughter cells are formed from this parent cell and both the daughter cells are haploid, right? Then both of these daughter cells are undergoing meiosis 2, and each daughter cells are making two daughter cells further, which are giving us total 4 daughter cells. So here we are getting 23 chromosomes only. This is a kind of equational division. So meiosis 1, if somebody asks you, meiosis is further divided into two types, meiosis 1, meiosis 2, which of these two is actually responsible for decreasing the chromosome number to half? So you have to remember it is always meiosis 1, right? Meiosis 2 is just like mitosis, it's just like equational division. And here I have given uh, an idea that you can understand that how the alleles are separated, right? So if you want to consider like this, I have drawn chromosome here. Okay, this is a homologous pair of chromosome. Here you can see two copies of capital T. On this chromosome you can see two copies of small t, right? This is the parent cell. This parent cell will divide and it will make two daughter cells, right? Up here I'll get capital T capital T, here I'll get small t, because there will be separation of homologous pair of chromosome, right? Then there will be separation of sister chromatids. So if let's suppose this is present like this and this is present like this, right? At last this one will receive capital T, only this one will receive capital T, this one will receive small t, this one will receive small t, right? So this is also very important to understand here, that during meiosis only separation of alleles take place, segregation of allele will occur in gametes because of the separation of the homologous pair and sister chromatid separation. I'll give a more idea about this at last, right? Now meiosis 1 we can study as under karyokinesis 1 and cytokinesis 1. Then meiosis 2 also karyokinesis 1 and sorry, meiosis 2 also has karyokinesis 2 and cytokinesis 2. In between meiosis 1 meiosis 2 there is a small duration phase that is called interkinesis, right? So let's start with meiosis 1 karyokinesis 1, and the very first one, that is prophase 1. A prophase 1 is different from prophase of mitosis, right? Prophase 1 is different from prophase of mitosis because it is very complex and very long as compared to normal prophase of mitosis, or because meiosis 2 and mitosis is exactly same, both are equational division, right? So instead of prophase of mitosis, if they are writing prophase 2 of meiosis 2, that also we can consider, right? It is further subdivided into 5 substages: leptotene, zygotene, pachytene, diplotene and dikinesis. These are the 5 substages of prophase 1. Okay, this is the cell which will enter from G2 stage into the meiosis 1 ka prophase 1, that is in the leptotene, right? When this kind of cell, you just try to see that it is having two duplicated centrioles, it's having nuclear envelope, it is having genetic material which is copied, or double chromatids sorry, or chromatin is copied there. Okay, here you can see in leptotene what will be the condition. In leptotene you have to remember there will be condensation of chromatin begins, right? So you will see something like chromosome will appear, but it will not look exactly like chromosome. Condensation will begin, it will continue until the chromosome become gradually visible under light microscope. This was a PYQ in 2024, same line, copy pasted, right? So this will continue until in the light microscope you can see gradually, slowly chromosomes are visible, right? It will appear exactly like this, right? Up here you can see nuclear envelope is present, nucleolus is also present, and endoplasmic reticulum Golgi complex will also be present in leptotene, right? Now once these chromosomes are visible to us, next stage is that we have to find which among these chromosomes are homologous pair, right? So what is the meaning of homologous chromosome? If the two chromosomes are exactly same in structure, and they are called homologous chromosome. They, if two chromosomes are called as homologous chromosome, they should have same size, they should have same position of centromere, and they should have same genes at the same position. Matlab, for example, if I want to draw homologous chromosome here, let's suppose I'm drawing two chromosome, right? You can see the size of these two chromosome is exactly same, the position of centromere is exactly same, the location or locus which I will select here, here or here, here, the gene which is present will also be same. So such kind of pair is called homologous pair, right? Here I have mentioned that, let's suppose this is a nucleus. In this nucleus you have two copies of chromosome number 1, two copies of chromosome number 2, two copies of chromosome number 3, two copies of chromosome number 4, up to chromosome number 23 you have two copies, right? So these both copies of each and every chromosome are considered one single homologous pair. So total, if we have 46 chromosome, how many homologous pair we do have? 23 pairs, right? So we have total chromosome as 46, but they are not 46 identical chromosomes. Actually these are 23 pairs of homologous chromosome, right? Now Same thing I have written here, that 22 pairs of these 23 are called autosomes, right, which are same in all the human beings. Matlab chromosome number 1 to 22 we all are having exactly same, but one pair is also called allosome or sex chromosome. This pair in female is homologous, which should be X and X, but in male it is non-homologous, it is not homologous. One chromosome is smaller, it is chromosome number Y, another one is chromosome X, right? So this is also one additional information. Okay, here I try to tell about these how these chromosomes will enter together. Let's suppose this is our parental generation. This was the chromosome in parent number 1, this was the chromosome in parent number 2. In the gametes only one chromatid will get delivered. When they will fuse, in G1 stage you will have this kind of condition, right? And once this G1 will enter into S, you will have these two chromosomes together in your single cell, right? So you can have a look, pause the slide and try to understand what I'm trying to explain here. I have added, like I have merged it with genetics concept so that you can clearly understand, right? Now from leptotene we'll come to zygotene. You remember in leptotene we were having some small visible chromosomes, right? So once the chromosomes are visible, we have to find out which are the homologous chromosome, right? Now every chromosome was here and there in the nucleus. Now what we have to do, the homologous chromosome must pair together. This is called synapsis or synapsis, right? So pairing of homologous chromosome should occur. Chromosomes must pair together. Pairing of homologous chromosome should occur. That is called synapsis. For this, there is a requirement of a protein complex which should be acting like a glue, right, by which both the chromosomes will come together. Now electron micrograph of this stage indicate that synapsis is accompanied by formation of synaptonymal complex, because actually protein complex, if you want to see, you need electron micrographs, right, because you cannot see them very clearly, right? Now synaptonymal complex, when you will see what is the synaptonymal complex, is a protein complex which is helping in keeping the homologous pair together. It acts like a glue. It provides the stability to the homologous chromosome, and pairing occurs because of this. So when one chromosome is paired with its homologous partner, it will form bivalent, right? What is the another meaning of bivalent? A pair of homologous chromosome. If a question is asked that formation of bivalent occur in which stage, so you have to remember it is zygotene stage. It will appear like this. See, in leptotene everything was looking like this, it was here and there. Here it has come together with the help of this synaptonymal complex, right? Bivalent formation has taken place here, or in simplified version you can see like this: chromosome number 1 is separated from chromosome number 1, 2 is also not present together, but here because of synaptonymal complex they have made a pair, that is called bivalent, two chromosome in one pair, right? What will happen, why they have come together? They have come together so that they can do crossing over, right? And crossing over will be done in packaging. Here you have to remember, in this stage still the chromosomes are not clearly visible, right? Sorry, still the chromosomes are not fully condensed, right? They are visible, they are not fully condensed. Condensation will continue, and in the packaging you have to remember that they will be distinctly visible. Chromosomes are fully like almost maximum condensed here, and that much condensed at least that you can see the chromatid, right? So let's suppose if this is one homologous pair which is having synaptonymal complex here, right, and they are paired up together, so you can count how many chromatids are there. I have labeled it as here 1, 2, 3, 4. So in a bivalent total, how many chromatids are there? 4, right? You can count the chromatids when the bivalent is clearly visible. If a question is asked that clearly visible bivalent or clearly visible tetrad is observed in which condition, so you have to remember it is observed in pack 18, right? So they have mentioned distinctly visible tetrad. Tetrad means 4 chromatid of one bivalent, or 4 chromatid of one homologous pair, right? So you have to remember this. Okay, now we are, why we are very much interested in knowing about this chromatid, because we need to find out which one is sister chromatid and which one is non sister chromatid. So you have to remember what is the meaning of sister chromatid. Sister-chromatid means chromatids attached to, okay, chromatid attached to single centromere. Matlab, if any chromatids are attached to one single centromere, it will be considered as sister chromatid. It means chromatid number 1 and 2 is sister chromatid, and chromatid number 3 and 4, they are sister chromatid to each other. But 1 and 3, 2 and 4, 2 and 3, 1 and 4, they are all non-sister chromatid. Okay. Now, no, no, sir. I think 30 minutes more. Sir. Okay. Now, you have to remember that here uh, crossing over will take place in between non-sister chromatid. So if this is 1, 2, 3, 4, right? So here you have to remember 4 and 1, they are crossing each other. These two are non-sister chromatid. These non-sister chromatid, they will cross each other, and there will be a formation of a structure which is called recombination nodule. Recombination nodule is a particular area or a site where crossing over has to be taken place, right? So here exchange will take place. This is called recombination nodule. At this recombination nodule, there will be activity of one a particular enzyme which is called recombinase activity. Recombinase activity will allow cutting, exchanging, and joining of the part of the chromatids, right? Now you can see, let's suppose this was first and this was fourth chromatid, right? So this is first and this is fourth. So this blue part will be cut, green part will be cut, it will be exchanged and it will resealed back. It will appear like this, right? So this will be recombined type of chromosome, right? Recombination has taken place here. अब you assume that if we label that here on this we have capital T gene, here you have capital T, here you have small t, and here you have small t, right? And if I can say that I cut it and I exchange it, so here the new combination will come as capital T small t, capital T small t, or not, right? Up now just think, if I'm doing the same recombination in between the sister chromatid, is there any use? No, because sister chromatids will have exactly same genes, right? If it is capital T here, it's copy of this, so it will be capital T here. If it is small t here, it is copy of this, it will be small t here as well, right? So there is no use of recombination in between the sister chromatid. The recombination must be done in between the non-sister chromatid of homologous pair of chromosome, right? Understood? So we have done the crossing over, right? Now if crossing over has been completed, there is no use of this synaptonemal complex to be there. The paired homologous chromosome must get separated, right? So there will be dissolution of synaptonemal complex in this stage, which is called diplotin stage, right? Bivalents will separate except at the place where they have done recombination. Can you see this stage, this site here? Crossing over has been completed, so at this place only the homologous pair will be sticked. Otherwise from every other region they will be separated because synaptonemal complex has been removed. This region will appear like a X-shaped structure when the synaptonemal complex is removed, right? This X-shaped structure is called chiasmata, right? Now in some of the vertebrates you have to remember that cells will be arrested at this stage. Cell will be arrested at this stage. They will not be able to complete diplotin stage, right? In the vertebrate cell, primary oocyte, okay, in the vertebrates primary oocyte is that particular cell which will be arrested in diplotin stage for several months to several years. So this stage is called prolonged diplotin stage or dictyotin stage. They have asked question on many times. Now dikinesis, what is dikinesis? Is the last phase of prophase 1. Here the spindle fibers are assembled together and they are ready to prepare the separation of homologous chromosome, right? Now complete terminalization of chiasmata will take place here during this stage. Nucleolus, Golgi complex, endoplasmic reticulum, everything, okay, it will disappear. It's just like last or late prophase, right? Spindle fibers here are assembled, okay, they are going to prepare the homologous chromosome for separation. Nuclear envelope is disappeared. Homologous chromosomes, they come in the cytoplasm, and now fully condensed chromosome, recombined chromosomes, they will appear like this. There is no chiasmata also. Now these chromosomes are ready to enter in metaphase 1, right? Now bivalents will come in the cytoplasm in the metaphase 1. These bivalents will align themselves in the center of the cell. Now this time, because bivalents are there, homologous pair of chromosome are present in the center of the cell, right? You will say there will be formation of a double metaphasic plate, right? Now the distribution of chromosome is completely random. Any type of chromosome, paternal or maternal, can be on any side, right? Now see here, this is the condition which will be there here. You can see recombination has achieved, right? Two lines of chromosome you can see here, right? And you can see to one chromosome how many spindles are attached, only one spindle, right? But here what you can observe, two spindles are attached to one chromosome and they are not recombined type of chromosome. This condition is there in metaphase and this is there in metaphase 1. This will lead to separation of homologous pair, right? This will lead to separation of homologous pair of chromosome in anaphase 1. The two chromosomes of each bivalent, they will start moving to two opposite pole. Matlab here who is getting separated? Homologous pair is getting separated. Is there any splitting of centromere? No, because to one chromosome only one spindle is attached, so there is no splitting in centromere, right? This result in the segregation or separation of homologous pair of chromosome or bivalent. Anaphase 1 is responsible for reduction in the number of chromosome. Can you see here? Okay, this was a pair, right? It got separated. The two homologous chromosomes are moving to two opposite pole. Now at each pole, can you see the chromosome number is going to reduce to half? Initially this particular cell was having how many chromosomes? This cell was having 4 chromosomes, right? Now when this will be packaged inside one nucleus and when this will be packaged inside another nucleus, it will receive only two chromosome or not, right? So we can say actually anaphase 1 is responsible for reducing the number of chromosome to half in the meiosis, right? An entire cell will have same number of chromosome, not doubled, right? Whereas in metaphase you can see it was different, right? Here it was uh, total 4. Okay, metaphase 2 it was like this, so in anaphase 2 it was like this. Okay, it was 8 here, it became 4 here. Okay, now in telophase 1, again this is the final step of karyokinesis 1. Here all the chromosomes which have reached two opposite ends will be packaged inside the nuclear envelope. They will reappear, right? Nuclear envelope will reappear. Golgi complex, ER and nucleus, these three will reappear. Chromosomes will uncoil, they will elongate, but they will not completely reach extremely extended state. They will not completely change into chromatin because again they have to enter in meiosis 2, right? So here this is a difference from telophase of mitosis. In telophase they will completely convert into chromatin. Here they will little bit decondense but not completely. They will enter into the you can say meiosis 2 for further division, right? So I have written this only. Now this results in the formation of two daughter cells. Both the daughter cells will receive half of the chromosomes, homologous pair separated. One centriole is present in each of the side of the cell, right? And then cytokinesis 1 will take place, right? And you will form two daughter cells. Now these two daughter cells have one problem, right? Now these two daughter cells have to enter in meiosis 2. For meiosis 2 they need two centrioles because meiosis 2 is exactly identical to mitosis, right? So for that we need to copy or we need to duplicate this centriole. This centriole duplication will take place in a very short lived stage, that is called interkinesis. In interkinesis it is also called intrameiotic interphase because it is coming in between the two meiotic divisions, meiosis 1 and meiosis 2, and it is a very short lived stage. In this stage only one thing is common to interphase, that RNA synthesis is there, protein synthesis is there. Okay, maybe centriole duplication is there, the most important centrosome or centriole duplication is there, but there is no DNA replication at all. This is the PYQ, always they will ask question, right? So now you have two haploid daughter cells which are formed with the two centrioles, right? Now they will enter into meiosis 2. Meiosis 2 always occur in haploid cell, it can never take place in diploid cell. Meiosis 1 always takes place in diploid cell, right? Okay, Now it is just similar to equational division, exactly same as mitosis. I have one slide where I have mentioned the diagrams of meiosis 2. I'll explain there. Only the nature of chromosome is recombinant. This is the difference. In mitosis, chromosomes are not recombined. Here, because meiosis 1 के बाद or followed by meiosis 1, meiosis 2 is coming, so the nature of chromosomes are recombinant. Otherwise, all the stages are similar. Both the daughter cells of meiosis 1 divides at each, and each cell is going to produce two daughter cells, total of four daughter cells, and all the four daughter cells are haploid, or we can say we have tetrad of cell, right? So here you can see this is the prophase 1, metaphase 1, anaphase 1, telophase 1. After this, they have not written cytokinesis 1 directly, they have moved to prophase 2. Can you see it's exactly like mitosis, right? You can see condensed chromosome, fully condensed. Now they will be aligned in the center, but the nature is that they are recombined. Then you can see splitting of centromere has taken place, and the sister chromatids are going to two opposite poles. And here you can see each of the daughter cells are receiving one chromatid, right? So if you try to see, if here you have total 2n equals to 4, right? Can you see here you are getting n equals to 2 only in each and every daughter cell, right? And here in this daughter cell also you have n equals to 2. Okay, so I hope this you have understood. Now this is the significance of meiosis. You have to remember, formation of gamete के लिए meiosis is very important. Diploid organism will always make gamete by meiosis only, and meiosis 1 can never take place in haploid. Always remember this, right? It will maintain the chromosome number. Meiosis will be responsible for decreasing the chromosome number to half, and then fertilization will again restore the chromosome number to original. Variations will be introduced because of meiosis only, because of this crossing over and the two random crossed over gametes fusing together, right? So fertilization, crossing over, both are very important for variation. And now we have to discuss change in amount of DNA and change in number of chromosome, which we have already completed, right? But we have to do one more thing by which we can make sure that yes, we have understood this process, right? So let's suppose if there is one parent cell which is in G1 stage, and I'm saying that this parent cell in G, or yeah, it is having, let's suppose, 1, 2, 3 and 4 chromosomes, right? So here n, if I will write, it is equivalent to 4, and if I will write C, it is also equivalent to 4. Any problem with this? I think no, right? After S phase, right, there will be one daughter cell which will be formed. This will be the case, right? This is G2 phase. In this, n equals to 4 only, but C has become 8, right? After this, what you have to remember, let's suppose this will enter into meiosis 1, right? After meiosis 1, this homologous pair will get separated, this homologous pair will get separated. So we'll be making two daughter cells here, right? One will receive this blue chromosome, this one will also receive blue chromosome, and one of this yellow chromosome. Correct or not? Because this pair separated, delivered into two daughter cells, this pair separated, delivered into two daughter cells. So after F1 in daughter cells, we can say N is equivalent to 2 here, and C is equivalent to 4, right? Then further they will divide. Now there will be separation of sister chromatids, right? So each and every daughter cell will be receiving one chromatid each or not, like this. So in the product, of meiosis 2, you will see N will be equivalent to 2 only, right? But C also will be equivalent to 2, right? So can you see that here there is actual change in the number of chromosome, right? Can we say this is reduction division, right? But if you see meiosis 2 and meiosis 1, can you see this is just like equational division? Just like the normal mitosis, the chromosome number is not changing at all. And if you see the amount of DNA, so it is from 4C, it has become 8C to 4C to 2C, right? Or if we start this with one you can say uh, DNA, then it will be 1C, 2C, 1C. Okay, we can write like this: 4C, okay, 4C, 8C, again 4C, okay, and here 2C. Okay, this is what? This is G1, this is G2, this is M1, and this is M2. Sometimes they give a question that the amount of DNA is 8 picogram in G1. What is the amount of DNA in M1, right? So if it is 8 here, it will become same here, 8 in M1, right? You can consider like this: if they're writing 2C here, it will become 4C here, it will become again 2C here, and it will become 1C here, right? So this pattern you have to remember. Okay, it will double here, it will decrease here, and it will again further decrease here, right? So you learn this is needed for solving all the questions, right? Up, we'll do these final questions and we'll wrap up, right? Here bivalent or tetrad formation is a characteristic feature observed during, in which you will see tetrad and bivalent formation is occurring, right? It is occurring, observed during chiasmata in zygote stage, bivalent tetrad formation, okay. Synaptonymal complex in zygote stage, chiasmata in diplotin stage, or synaptonymal complex in pachytene stage. So you will see that the first step was leptotene, then zygotine, then pachytene, then diplotin, then what was the last one? Terminalization of chiasmata occurs in dikinesis. So here formation word is used, distinct formation is not used. So formation, you have to remember, it will occur in zygotine, right? So in the same stage where synaptonymal complex will be formed, in that stage only this will be formed. The appearance of recombination nodule on homologous chromosome during the meiosis characterizes what? Sites which, on which crossing over occurs, right? The site at which crossing over has occurred is chiasmata. The site where the crossing over occurs or will take place, that is recombination nodule. Regarding meiosis, which of the following statement is correct? Sorry, incorrect, right? 4 haploid cells are formed in the end of meiosis 2, correct. There are 2 stages in meiosis, meiosis 1 and 2, yes. DNA replication occur in S phase of meiosis 2, this is wrong, right? DNA replication will never take place in meiosis 2, it will take place in interphase, right? Pairing of homologous chromosome and recombination occur in meiosis 1, this is correct, right? Which stage of meiotic prophase shows terminalization of chiasmata as its distinct feature? Chiasmata will get terminalized at dikinesis, last, right? Last stage. Okay, which of the following stages of meiosis involves division of centromere? Centromere will split. Okay, centromere will divide in which stage? In anaphase 1. Even centromere will divide in anaphase also, but it will never divide in anaphase 1, right? This can you do? Zygote in what will be the thing which you have to remember? There will be synapsis, right? What about pachytene? Pachytene mein crossing over will take place, right? What about diplotin? Diplotin mein you have to remember there will be dikinesis, there will be termination of chiasmata. Pachytene, there will be crossing over, right? So which one is left here? 2, just a minute, okay. In zygotine there will be synapsis, okay. In pachytene there will be crossing over. In diplotin there will be visibility of chiasmata, and in Dikinesis, there will be termination of gas matter, right? So 4, 3, 2, 1, first option. Okay, so this is all for today. We have completed revision of plant growth and development in detail. We have completed a revision of cell cycle and cell division in detail. If you have any kind of questions to be asked, you can write it down in the comment section, and please study very hard. Very few days are left. Please revise everything. Please be motivated, be positive. Thank you. All the best.